Seattle Cell Symposium today. Good morning to our West Coast colleagues, and good afternoon or evening to those of you joining from elsewhere. My name is Megan, and I'm a member of the Allen Institute Events Team and the Seattle Cell Symposium Planning Committee. Before we get started, just at 1/2 the hour, I have just a few housekeeping notes to cover before we get started. First of all, we'd love to know who is here with us today. So please take a moment to introduce yourself in the chat, send your name, affiliation, and what you're excited to learn about today. Be sure that you are sending chats to all panelists and attendees. In that chat window, you can select all panelists and attendees from the drop-down window. Again, good morning, afternoon, or evening to those of you joining us. Please take a moment to introduce yourself in the chat and let us know what brought you here today, why you registered for this event. As those of you continue to introduce yourself in the chat, I have just a couple of more notes to share. When we do get started, please be sure to send any questions to our presenters in the Q&A panel, also accessible from your Zoom toolbar. Be sure to include the speaker's name to who your question is directed, and be sure to hit send from that window. We have tried to save time for questions at the end of each presentation. If we don't get to them during the live Q&A, our speakers will be able to type their answers afterwards. And for those of you joining us on YouTube, we will be looking for questions there as well. So please be sure to submit your questions in the YouTube live chat. If you are having any connectivity issues, we do have a couple of options. The first is you can view the event stream on YouTube by clicking a link we will add to the chat window. You can also select from your from Zoom the view stream on custom live streaming service. You are also welcome to call in from your phone for better audio by clicking to the switch phone audio option from your audio menu. Here's a brief overview of today's agenda. We will have some introductory remarks, followed by presentations, and concluding with a panel discussion with all presenters. We do hope that you can stay for the entire program, but if you do have to step away, we will be posting a full recording of the event on the event website and our YouTube channel after the event. Throughout the day, our event will be moderated by other members of the Symposium Planning Committee, who have worked together for months to help plan this program and the details for today and tomorrow. And as a reminder, this is day one. We will be hosting a second half day of presentations tomorrow, starting again at 8:30 a.m. Pacific time. Let's see, we have Tim from Journal of Cell Biology. Welcome. Welcome, Angela from the Netherlands. Joe from UCSD, one of our speakers. Sharon from Duke. Welcome. Again, to those of you who are just joining us, we are going to get started shortly, but please feel free to take a minute to introduce yourself in the chat. Let us know your name and what you're looking forward to hearing today. Welcome, June from UC Irvine. Excellent. So we're just at 1/2 the hour. So I am excited to introduce Executive Director of the Allen Institute for Cell Science, Roo Gunawardena. Welcome, Roo.
Thanks, Megan. Hello, everyone, and welcome to our annual Cell Science Symposium. This is the second year in a row we are hosting this virtually. But we are excited that this allows us to reach a wider audience and also a wider range of speakers today. So first, I want to thank this amazing group of people, part of our Cell Science Symposium committee, who worked really hard to put this exciting program together. So thank you, guys. And I also want to take a second to thank the entire institute, the team, for all their hard work and dedication over the last year, another year of the pandemic, to work hard on delivering on our mission. So today and tomorrow, 2 half days. So the symposium theme is around data integration, from multiomics to imaging. So this is a topic that is also a focus of our own work at the Cell Institute, as you'll hear today and tomorrow, but also a big challenge in the field on how to put data together to gain insight. So we have a great lineup of speakers who are going to touch on this topic and share how they're addressing this challenge around a variety of cell biology topics. So I'm very excited to hear them and hope you are too. I hope you enjoy the talks and also find it informative. In addition to that, we're going to have some introductory remarks both today and tomorrow about our institute, and 2 team talks from our staff, and 2 panel discussions like Megan just discussed to delve a little bit deeper into this topic with our speakers. I hope you enjoyed that as well. So with that, I want to thank all of you for attending our symposium and hope you enjoy it. And with that, I'm going to hand this over to Susan, who's our Deputy Director at the Institute, who'll give a few introductory remarks about what we do at the Institute and our research programs. So thank you. And Susan.
Thank you, Lou. So just making sure I have control here. I did click. There we go. Great. So before we get started, I'd like to share more about the mission and approach of the Allen Institute for Cell Science. But before I tell you what we do, I do want to introduce you to who we are. We're a fun-loving team of about 60 people who have been working together for about 6 years now. Even as the landscape of how we work together has changed, our focus has continued to be on our core values of open team science and community engagement. Now, on to what we do. At the Allen Institute of Cell Science, our scientific mission is to understand how cells organize themselves and change. We believe that the key to understanding the cell is to understand the interplay between how cells are organized, their function, behavior, and their identity. And we really need to understand how these interplay in time. If we do that, we believe we should be able to, all of you, should be able to look at a cell and know what it is doing, what it did, and what it will do. But how are we going to do this? So cells have to coordinate all of their intracellular processes over 4 orders of magnitude to achieve their cell level behavior. Our goal is to develop a standard model of cell organization and cell state transitions in differentiation and disease. And to achieve this goal, we have focused in on studying the cell at the level of intracellular structures or organelles. And this permits us to maintain a global view of the cell while tracking many fewer types of components, 30 to 50 perhaps, instead of all the proteins in the cell, to permit us to make the cell's complexity a little more humanly interpretable. Other hallmarks of our approach is that we want to consider the cell as a whole, and we want to understand normal cells to understand what it means to be abnormal. So we have really invested in the concept of normal in addition to the concept of perturbation. We focus in on 3D and on generating approaches that will permit us to embrace those dynamics of the cell. And we do all of this by using quantitative methods. But we know that this is not sufficient to truly understand the whole cell. We also integrate our approaches with the approaches developed in the larger cell science community to bring together our 3D live cell imaging with other data types, such as expression signatures, protein states, and chromatin organization. And that's also why we're so excited today for the theme of the symposium and really highlighting all these ideas and concepts out there to integrate and come together to understand the cell. We at the Allen Institute for Cell Science, and I think many of you in this audience, are excited about the prospect of single cell imaging and analysis being the next step in the post-genomic era. But as we consider these amazing images of cells, we face a grand challenge. How do we establish a quantitative framework to compare the shapes and positions in 3D for cellular structures and how they change over time, when the concepts of how to measure cellular structures are not even themselves straightforward? These are the types of approaches that need to be developed and tested and iterated upon. A second part of our mission is to develop and democratize tools to discover new biology together as a community. And we can try to address this grand challenge I just stated and work towards both parts of our mission by creating accessible new methods and tools to perform accurate, quantitative, robust imaging and analysis at large scale to extract quantitative cell organization measurements. All that we do and develop is accessible and available at our web portal, the Allen Cell Explorer. Please check us out at allencell.org. Now, I'd like to take a moment to walk you through our scientific programs and projects to provide a little bit of context for the two talks that you're going to hear about today and tomorrow at the symposium, and to learn a little bit more about us. Our model system is the human-induced pluripotent stem cell. As such, we study these cells in their pluripotent state, focusing on what stem cells do best, grow and divide, to study how the organization inside these cells is generated and regulated. And then we study the other thing that these cells do best, which is to differentiate, in our case, along the mesodermal lineage into cardiomyocytes and endothelial cells. As I already said, we also have our dissemination mission, which we also address in part with our data flow program to develop and democratize tools and to make all of this work more feasible. And Graham will tell you more about that tomorrow. So first, to study how organization inside the cell is generated and regulated, we have two approaches. We want to go broad, testing our goal of a holistic view of the cell and how many structures in the cell come together and are organized in it. For this goal, we've worked towards several methods to integrate structures in cells. Most recently, we've performed a large-scale analysis of 25 cell lines and developed a quantitative framework to study the locations, amounts, and their variation within the HIPSC as a normal baseline for cell organization. We found that integrated intracellular organization is extremely robust to changes in cell shape. Our second approach here is to study individual structures and start to incorporate those dynamics and create a quantitative workflow to handle them. And here we've started with the nucleus and nuclear growth and shape regulation, which you will hear more about in tomorrow's cell science talk. We've also teamed up with the 40 nucleome and the wider community to study the integrated organization inside the nucleus, where now we have the challenge that we need to integrate this organization with that of the genome. Here, we want to embrace the challenge of doing this integration in a way that incorporates the holistic shape of the nucleus and its variation within cells. And so for that project, we really come together with our 40 nucleome community and do a very large data integration effort that really spans many multi-omics to images. Within our differentiation program, we initially focused on differentiated cardiomyocytes and their internal organization. Last year, we published a study addressing the question whether transcript abundance can predict cell organization using these cardiomyocytes as our model system. We created approaches to quantify cardiomyocyte sarcomere organization, as well as gene expression by Rna Fish, and developed analyses to integrate cell organization and transcripts in single cells. We found that gene expression cannot predict structural organization at the single cell level. We've paused our work with the cardiomyocytes to focus in on the earliest stages of state change as the pluripotent cells begin their differentiation. We find a reproducible, consistent EMT-like transition as cells change from epithelial-like to migratory, a project you will hear much more about later today in our cell science talk. We're excited to move into adding yet another cell type to our model system, the differentiated endothelial cell, and further to eventually explore those intermediate states between these parts of the differentiation process. We're also focusing in on how to study cell-cell interactions within our cell colonies, which is really part of both of these programs and everything that our cells do. And we really importantly recognize that the tools and approaches between these two scientific programs are very greatly interlinked and can inform each other. And we're excited to start taking our approaches that we developed in the pluripotent state and take them into our state-changing differentiation part of the Institute as well. So with that, I'd like to wrap up this brief overview of our Institute science with a great thank you to everyone on the team and really move to start this exciting symposium. Here's a snapshot of the agenda today. We have a fantastic lineup of presenters, and we'll end the session with a panel discussion. So now I'm happy to introduce our first speaker of the day, Dr. Lucas Pelkemans. Dr. Pelkemans is a professor at the Department of Molecular Life Sciences at the University of Zurich. His lab studies the information processing capacity of human cells during growth factor stimulation using multiple markers of cellular state. Today, he'll be giving a talk titled Multimodal Perception Links Cellular State to Decision-Making in Single Cells. Please welcome Lucas.
All right, thank you very much. It's great to attend this. Here it's actually evening right now. I think it's early morning where you are. So anyway, I'm excited to give a talk. I will talk about something that some of it has actually been on bioRxiv since quite a while. The more recent stuff is now in revision, so some of that is probably not visible in the bioRxiv paper, but I'll walk you through it. Let me see, I can go, yes, great. So, you know, I first of all, I think, obviously, you know, imaging across scales generates a lot of beautiful images, and one can appreciate that. I will not show that many images. That doesn't mean we don't have lots of images, but I really want to take sort of the step, one step further and ask, you know, what do we do with these images? What is the information we can extract out of it? And what can we do in terms of using information and understanding biology better? And I like to sort of put some points here on this slide, which I appreciate a lot from people from information theory, who, you know, often like to say that, you know, to them, information is to biology what energy is to physics. And so just to sort of point out some I think the capacity to store and process information may be the most fundamental and distinctive property of living systems compared to non-living systems. Life's ability to structure matter and make it functional via manipulation of information is very unlike what we see in any other kind of physical system. And also, and this is obvious, right, in living systems, information is, of course, physical, right? A change in potential or a post-translational modification, and it can be measured. So, you know, like energy may be somewhat of a vague concept sometimes in physics, it's also a very concrete concept, and I think one can very much see the same in biology. To some extent, it's vague to talk about information, but on the other hand, it's very concrete and physical, such as, let's say, the phosphorylation of a protein. So, you know, I want to sort of bring in a few concepts before I start talking about our results, our latest results. And I want to introduce two concepts that really come from information theory and from the study of perception, many of which has actually been done in neuroscience. But this will definitely not be a talk about neuroscience. It will be actually a talk about single cells. But some of the concepts that have been well developed in neuroscience, I think, are also applicable to how single cells process information. So first, I want to just quickly introduce multimodal perception. This is something that we are all, I think, very familiar with, and it basically tells or a concept that we use multiple modes, right, like our sight, our smell, our touch, our taste, to create a more comprehensive picture of the world around us. And in a way, we sort of think about this in two ways. So there is partial redundancy in how different modes give us information, and that redundancy gives robustness because you are insensitive to uncertainties in individual signaling inputs. But there's also a non-redundant element in this, and that can give comprehensiveness. It allows you to integrate multiple sources of different information. The second point I want to quickly highlight is contextual information processing. So obviously here you see 2 times exactly the same shape, but our brain, right, our neurons, our neural network interprets that in a contextual manner, such that you read this as the cat, and therefore these 2 identical symbols are once interpreted as an H and once as an A. And this is what we do all the time, right? Human perception is contextual. And so that gives this, that allows us to, or that basically the result of that is that the same H can give a different perception depending on the context, or sometimes different inputs can give the same perception depending on the context. So does this apply to single cells, in particular single mammalian human tissue culture cells? Well, there's a lot of analogies you can. So first of all, if you think about cellular information processing, typically this is done by relatively complex networks of signaling nodes, as sort of illustrated here. And it's through that network through which cells sort of try to process information about the amount of growth factor they are being exposed to. But we also know, and I think Susan very nicely introduced that, right? The cells have, of course, their cytoskeleton, their organelles, their proliferative states, their metabolic states. And we know from many papers in the literature that this influences signal transduction quite extensively. Now, classically, it's often studied in the context of thinking, look, you know, endosomes could be a nice platform on which you can allow two signaling molecules to meet, and that gives you sort of the mechanism by which you can transduce a signal. But if that happens like that, and there are of course many examples where it happens like that, it also gives signaling the opportunity to sense the abundance of endosomes in the cell, right? Because if there are more endosomes, you have more of the platform, you might get more efficient signal transduction than if there are less endosomes. And of course, this is sort of generalizable across many of the cellular structures we have in a cell. So sort of one of the main questions we postulated is, could the cellular state or all these properties of the cellular state be the source of heterogeneity that we see in signaling responses between genetically identical cells? Because if we highlight, let's say, a typical signaling node in a network, and we look at how individual cells respond to a certain input of growth factor, a certain concentration of growth factor, you see that there are very broad distributions of individual cellular responses that highly overlap, right? In this case, we are looking at phosphorylated levels of MEC, and you see that these distributions highly overlap. So in other words, right, does this heterogeneity reflect uncertainty, which many people have thought in the past, right? Because you see so much overlap. So in other words, an individual cell doesn't have a good capacity to really distinguish between different levels of EGF. Or does it actually reflect contextual information processing? And if it would be that, then maybe it would actually have a lot of capacity to sense such kind of information or integrate such kind of information. But what we have been missing in the past is to actually understand in which context cells are doing that. Now, there is, of course, the problem of feedback if you want to study this experimentally, right? Obviously, the cellular state influences signal transduction, but signal transduction will continuously update your state, right? Downstream of signaling, you get changes in your cytoskeleton, changes in your organelle positioning, etc. And that, of course, is a bit tricky if we want to study sort of the direction of causality. However, one way to sort of experimentally overcome that is to, just do very classical starvation of your cells, serum starvation, growth factor starvation, by short induction, right? So typically, right, we would do a 16-hour growth factor starvation, and then we just do a 5-minute pulse of EGF. And then you can test whether your state properties of your cells are actually changing in the 6-minute pulse of EGF. And typically, and certainly all the properties I will show today, are not changing in that short time scale. So we can then ask experimentally the question, does pre-existing heterogeneity in cellular states determine the heterogeneity in acute signaling responses? Because after starvation, the cells are not active in signaling, that's very clear. And then after 5 minutes, they become active. And then you can ask, is the heterogeneity you see after those 5 minutes pre-existing, right? Or correlated, determined by the pre-existing heterogeneity in these cellular state properties. And to just give you, a visual view of that, right? If you look at, these are typical human tissue culture cells grown in culture, and we stay in here for 3 cellular state markers. And this is a typical image where you see pre-existing heterogeneity in cellular states. Now, these same cells were now exposed for 5 minutes of EGF, and this is the heterogeneity you see in typical signaling response markers. And so the basic question is very simple, right? Is the heterogeneity you see here on the right side somehow related to the heterogeneity we see on the left side? So for that, we use methods that we developed in the lab in the past. One is 4i, which basically allows us to do quantitative multi-scale multiplex imaging, which we combine with computer vision and AI. Then in this study, we decided to look at 10 signaling response markers in the same single cell. So you see them sort of listed here and numbered here. And we also decided to look at 20 cellular state markers, which cover a range of different properties of the cells, many structures and organelles, but also metabolic states, proliferative states, the cell cycle, and things like that. So let's focus for a moment on the cellular state markers. So we stain all these markers in the same single cells, and then using computer vision methods, we extract a total of 650 features from multiple scales. The multicellular scale, the cellular scale, and the subcellular scale. So we look, for instance, at neighbors of cells, their contacts, local cell crowding. These are sort of the multicellular scale properties. We look at cell size, cell shape features, abundances of certain markers. These are cellular scale properties. And then we can look at subcellular shapes of organelles or textures or other types of subcellular features. What I just show you here now is sort of we call this the cellular state landscape. It is now a highly reduced view of that multidimensional space, right? It's just basically we converted those 650 features in, I think, about 150 principal components, and we use those to then project that in a two-dimensional UMAP. So it's highly reduced, but it just gives you sort of the overall view of the landscape of single cells in that population. And the point I want to make here is that when you look at cells that were just starved, so that is 0 nanogram per ml EGF, or cells that were exposed to 100 nanogram per ml EGF, all of them mix very well across this landscape. So this is a cellular landscape which exists in just starved cells as well as in cells exposed to 5 minutes of EGF. So in other words, it's a pre-existing or non-changing cellular state landscape. Just to highlight a little bit what the cellular landscape shows, it basically shows you different, gradients of individual features. If you overlay those, a typical one is, for instance, DAPI, where obviously this island here, which is in red, are of course G2 cells, and this bigger island here in bluish are G1 cells. And then you can have abundance of mitochondria, of ER exit sites, of mechanical state through YAP1, subcellular features, etc. Okay, now having this pre-existing or non-changing cellular state landscape, we then can ask the question, do now our signaling responses, which are also heterogeneities, actually follow a certain pattern or show a certain pattern on this landscape? And if they do, it would already suggest that this is not just random variation in signaling responses, it may actually be very much determined by the cellular state landscape. And that is indeed what we see. So if we look again here at 3 of these signaling responses, you do see that they form clear gradients across this cellular state landscape. The gradients are not the same, they're somewhat similar, they're somewhat similar and somewhat dissimilar. And I will come back to that in a moment. Now, interestingly, what you can then do once you have all these non-changing cellular state features, you can build models. You can even do this in relatively simple ways using multilinear regression. And we can ask, to which extent can we actually predict the levels of these signaling responses in single cells using those non-changing cellular state features. These are the predicted values overlaid again over the landscape, and you can, I think, appreciate that this prediction is highly accurate. You can also visualize this by just projecting side-by-side actual segmented cells color-coded for the measured levels of phosphorylated ERG versus predicted levels of phosphorylated ERG, and it's very hard to actually see a difference. So to a large extent, the variation that you see in signaling responses is determined and can be predicted by properties of the cellular state. Okay, so now, right, having this, we can ask the question, okay, is the cellular state really giving the context by which individual cells interpret growth factor signaling? And so to sort of study that, what we did is the following. So we asked, how accurate, now knowing this, could single cells distinguish 5 concentrations of EGF? We're going to ask this in different ways and compare this in different ways. So the first way we ask this is what we call unimodal perception. So that's just one signaling node, and we assume the response is non-contextual. So in other words, we just take the levels of phospho-ERG in single cells. And we know from distributions that the levels of phospho-ERG look a bit like this. So you would already know or see from these types of distributions that it's probably not going to be very accurate in distinguishing different levels of EGF. It may distinguish 0 from 100 nanogram very well, but in between, it might not be doing a very good job. You can test this with some kind of classifier. You basically train a model using just levels of phosphoric and ask how well is the model able to predict that a single cell is exposed to one of those 5 concentrations of EGF. And that's basically shown in this kind of box plot graph where you have your classifier probability here on the y-axis, and then you see different amounts of EGF received. And for each amount we get probability of belonging to indeed the right class, right? So for instance, 0, pretty high probability, the classifier would say that it's indeed exposed to 0. But if you look here on the rest of the amounts of EGF received, it's really bad in predicting it accurately. You can also quantify this in the framework of information theory, which basically is relatively straightforward. You basically ask, what is the capacity of single cells to perceive 5 concentrations of EGF? And so if you're unimodal, or in this case using phosphoric and non-contextual, you have about 0.8 bits of capacity to perceive 5 concentrations of EGF. And 0.8 bits basically means you might be able to distinguish between a bit less than just 2 concentrations, right? 1 bit is 2 concentrations. So that's clearly not very good. The next we ask, what could cells distinguish if they, what we would call multimodal perception, so they actually use information from all signaling nodes in the network, but we still assume those responses are non-contextual, right? So we just take the signaling responses of all these 10 nodes. And when you look then at the accuracy of the model, it gets better, right? You see that the prediction accuracy gets better, but it still has some confusion here. And you can also appreciate that in the bits, it's about 1.2 bits. It's still not great. Okay, now let's ask what happens if we consider that this is a contextual signal. So you take into account the context of the cellular state in which each single cell receives the input. Or to put it very simply, we're basically asking, let's compare cells that are all in the exact same state, and are they now much better able to distinguish the different levels of EGF? So we take unimodal, right, phosphoric, but now contextual, so taking into account the cellular state. And indeed, compared to 0.8, we are now to about 1.3. And you also see that in the box plot here on the left, the box plots are getting close to the right probability, or high probability for the right class. It's still not great though. If you now, however, take multimodal perception into account, so taking information from all signaling nodes in contextual to the cellular state, we actually realize that now they get close to perfect perception. So perfect perception is the dashed line here on the top, which would be bits, 2.3 bits, which is basically allowing you to perfectly separate between 5 concentrations of EGF. So by adding the contextual information, right, we realize that cells actually have a much better, or in principle, could have a much better ability to distinguish growth factor concentrations. One point to make, right, just for sort of sanity check, if you just use context-only features, they're of course pre-existing, they shouldn't change upon adding EGF. You should not have any predictive power, and that's indeed the case. You basically are close to random perception, so not having any ability to distinguish. Okay, so that gives us the idea that maybe indeed cellular signaling is contextual, and maybe cells use multimodal perception, so the information of multiple signaling nodes, to do a very accurate perception of the amount of growth factor they are exposed to. So when you think a little bit about this for a while, actually the real question sort of that, or one of the questions that came up to us is, well, maybe that actually points out to us that cells have complex signaling networks, not only to perceive the amount of growth factor they're exposed to, but also, and perhaps even primarily, the state they are in. And some sort of, results around that were really sort of inspired by this kind of thinking. So I just showed you before that, you can have a signaling response, right? That is determined by a certain cellular state or a certain set of cellular states. And you could imagine in a totally redundant hypothetical case that you would have 3 cellular states and you have 2 responses, and each of those 3 states activates each of those 2 responses. Now, if that would be the case, then of course, either individually or together, A and B cannot distinguish between the states, right? They cannot give any information about being in either state 1, 2, or 3. And what we say is response A and B therefore contain redundant information about states. You can also think about it in this way. Let's imagine there's a partially non-redundant configuration where you, for instance, have again these 3 states, but state 1 and 2 activate A, 2 and 3 activate B. So there's some redundancy in A, right? A is redundant about 1 and 2, right? While B is redundant about 2 and 3. But together, right, they can actually distinguish all 3 states. Because as a multimodal response, A and B distinguish them all. If only A is on, you must be in state 1. If only B is on, you must be in state 3. And if A and B are both on, you must be in 2, right? So that is how partial non-redundancy can give you a more comprehensive ability to sense, in this case, the number of states you are in. So we call this partially non-redundant information about states. So from our analysis I showed you before, some pieces of information suggested to us that this may really be the case. So first of all, we asked the type of variation in our data. So in other words, if we compare the amount of variation that is sort of induced or caused by differences in cellular state, which is here on the y-axis, compared to the variation induced by changing factor concentration, we typically actually see that cellular state causes more differences in signaling responses than changes in growth factor. So in other words, a lot of information in signaling responses is about the cellular state than about amount of EGF. And second, if we ask from all of the models we learned to predict each of these signaling responses using cellular state markers, and we ask which of the cellular state properties were most important for predicting the responses of signaling nodes, we realized that we get signaling node specific patterns. So nodes are not capturing the same information about the cellular state. They capture, when you look at this picture, somewhat partially non-redundant information. Maybe I can visualize this even a bit better with these plots. So let's take 2 very well-known signaling responses, phosphor-ERK and phosphorylated mTOR. They correlate very strongly with each other, right? As you can see in this plot. But what we now did on top here, we overlaid, these are all single cells, single dots, and in the same single cells, we plotted the amount, z-scored here, of the abundance of Sec13, a marker of ER exit sites, or the amount of HSP60, a marker of mitochondria. And I hope you can appreciate that the gradient going from red to blue is here along the y-axis, while here the gradient is actually along the x-axis, right? So if you would do a partial analysis, that's what partial analysis is, you can ask, if we increase Sec13 and we look at how phospho-ERK or phospho-mTOR respond to that partial to the other marker, we see that only phospho-ERK responds, right? Because most of this variation is along this axis, right? While for HSP60, the partial analysis shows that phospho-mTOR is the one responding. So it's more along this axis. So again, there's from information theory, there's ways of sort of quantifying that, and that allows you to quantify how much is there redundant information between these signaling nodes about, in this case, Sec13 or HSP60, and to which extent do signaling nodes carry unique information. And then sort of gives the same picture, right? They, of course, both have redundant information, but phospho-ERK has unique information about Sec13, while phospho-mTOR has unique information about HSP60 or mitochondria. So this already shows us that indeed, signaling nodes have partially redundant and partially non-redundant information about cellular state properties. So we then decided, okay, then cells use this, right, using multiple signaling nodes to actually capture or know comprehensively what is their cellular state, or in other words, to put this in a sort of a statistical framework, could cells know their position in cellular state space by using their signaling responses, right? So in other words, we ask this as the following. We look at a single cell, this green cell, and we look at its direct neighbors in cellular state space, multidimensional cellular state space. And then we ask for that same single cell, that green single cell, what are its neighbors in signaling space? And we can take, let's say, a unimodal signaling space, so let's say just based on phospho-ERK, or a bimodal signaling space, phospho-ERK and mTOR, or a multimodal signaling space, all the 10 signaling nodes. And we ask, looking at those neighbors and looking at the neighbors of these guys, to which extent are they the same, right? And do we perhaps see that if we take information from multiple signaling nodes, the neighbors are actually going to overlap more, which would mean that indeed, based on the multimodal signaling response, that gives enough information to cells to know where in the cellular state space a single cell is sitting. So you can overlay this kind of quantification again on the cellular state map. And here you see how good cells are able to know their position based on just phospho-ERK. And they know it for some regions, but only for some, right? In this region, it's actually pretty bad. Phospho-mTOR actually gives information about other regions on the cellular state space map, right? It's better here and here, which is quite different from what the information that phospho-ERK gives. Together, they actually synergize and give already quite good information about your position in cellular state space. But if we take the multimodal signaling response, we actually realize that now cells have a very comprehensive picture of their position in cellular state space. So, I mean, this is another way of sort of showing a similar analysis. We can take the cellular state space, do some kind of fuzzy clustering to cluster single cells in different now discrete groups of cellular states, in this case, 18 classes of cellular states, sort of color-coded here on this projection. And then we can ask, how well can we predict this state, right? So we have true state classes and predicted state classes by knowing just phospho-ERK or phospho-mTOR. You see that this is really not so good. mTOR is a bit better, but still not very good. If we take both, we get already better, still not perfect. And if we take the multimodal signaling response, we start to have mainly accurate prediction, meaning along the diagonal. You can quantify this also in terms of information content, and then we realize that this has at least 3.4 bits of information, which at least based on these measurements would allow a perfect decoding of about 11 cellular states. Good, so this is mainly what I wanted to show. It's really more about the question, how do cells, having all their cellular state properties, their organelles, their cytoskeleton, and how this varies between single cells, how do cells actually capture that information, and are they able to capture that information comprehensively? So to give a little summary on that, I hope I've been able to show you that including multiple cellular state markers when measuring signaling responses, which is not something people do these days actually standardly, right? Most people who study signaling are just looking at signaling, right? Or multiple signaling markers. But I would argue that you really also need to include multiple cellular state markers to actually understand your signaling responses. And it basically shows that signaling heterogeneity contains quite a lot of additional information and doesn't reflect uncertainty. I hope I've been able to show you that the concepts of contextual information processing and multimodal perception that we know of that is performed by multicellular networks, like our brain, also apply to information processing by molecular networks, right, inside single cells. So I think that's sort of that concept can be extended to also information processing inside single cells. And this framework reveals predictive rules that link information about the physical chemical state of individual cells to decision-making and reveal which properties are most relevant. And I didn't show you today, but that's really for a lack of time, whether, because, it's obvious that all that I've shown you, you could ask the question, do cells actually utilize now this information to make a decision to differentiate into a certain direction or to re-enter the cell cycle, things like that. And indeed, we are able to see this now that, for instance, looking at re-entry of the cell cycle, it is based on the cellular state, you can start to very accurately predict at the single cell level which cell will re-enter the cell cycle upon, for instance, growth factor stimulation. So with that, I will wrap up and acknowledge my lab, picture of my lab this summer, the funding here indicated, and thank you for your attention.
Great. Thank you for a great talk, Lucas. And hello, everyone. My name is Gökhan Dalgın. I am a senior scientist at the Allen Institute for Cell Science. I will be moderating the Q&A for the first few speakers. How about, Lucas, we have multiple questions in the Q&A panel, and let me ask one of them, and the others, if you can type your answers in the Q&A, that will be great.
Sure.
Prasun from Prasun has asked about how then biochemical assay like Western blot can catch those dependent responses.
Right. So, well, if you take a Western blot, basically look at the mean shifts, right? So the mean shifts of your whole population, and they of course are there, right? But if you look at the single cells, you get a broad distribution. So it's very easy to, even though you can nicely separate mean responses, that there are single cells in your distribution that would actually overlap fully with single cells of another distribution that is exposed to another concentration of EGF. So Western blots, yes, that's of course was the technology in the old days, but Western blots give you a very confusing picture because it only gives mean response, and if you have bimodal responses or things like that, the mean doesn't really mean much, right? So that would be my answer.
Thank you. Again, there are a couple more questions if you can type them in the Q&A.
Sure.
And thank you again, Lucas.
You're welcome.
Now we are to our next presenter, Dr. Chantel Evans. She's an assistant professor of cell biology at Duke University. Her lab uses advanced microscopy and biochemical techniques to gain insight into molecular mechanisms that regulate mitophagy in primary neurons. Today, she will be giving a talk titled Investigating the Spatio-Temporal Dynamics of Neuronal Mitophagy. Please welcome Chantel.
Thank you very much for that introduction, and thank you for allowing me to be here today to share my research with you. So what I'm going to talk about today is actually from my postdoc when I was at the University of Pennsylvania in Erica Holzbauer's lab. I've recently transitioned to running my own lab at Duke, and so a lot of what we're going to focus on is looking at how we used live cell imaging and quantitative approaches to understand mitochondrial dynamics in the context of neurons. So neurons are highly polarized and terminally differentiated cells, meaning neurons must be maintained for the lifetime of an animal. And axonal transport is required for both the supply and clearance of proteins in organelles, as well as for spatiotemporal signaling. And due to this unique polarity, neurons rely heavily on mitochondria to support local energy demands at fusion sites that can be located far from the soma. So to put this into a better perspective, what I'm showing you here is a motor neuron drawn to scale, where this small red dot in the upper left-hand corner is representing the soma, and then this long continuous line winding back and forth is the axon. So I think from this depiction, it's pretty easy to see how important it is to know when and where a protein or organelle is within the axon, and how defects in these maintenance mechanisms could lead to the progressive functional and structural degeneration of the neuron. So damaged or aged mitochondria are removed from the cell via the mitophagy pathway. This is a constitutive pathway that initiates via oxidative stress, or in cells, we can induce that in cell culture using chemicals that damage mitochondria, such as CCCP or antimycin A. So following mitochondrial damage, you get the translocation of PINK1 to, you get the stabilization of PINK1 to the mitochondrial membrane. This leads to the recruitment of an E3 ubiquitin ligase, Parkin, and together these two proteins will start to phosphoubiquitinate other outer mitochondrial membrane proteins. This eventually leads to the translocation of optineurin and TBK1, which allows for the recruitment of an autophagosome, or a double membrane structure that sequesters this damaged organelle. This autophagosome can then fuse with lysosomes, where lysosomal enzymes will start to degrade that specific organelle. And today we're going to focus on this protein optineurin, which is an autophagy receptor for this mitophagy pathway. It basically acts as a bridge because it can bind to the ubiquitin that's located on these damaged organelles, as well as binding to LC3, which is on that growing autophagosome. And so if you look in the literature, mitophagy has been primarily studied in HeLa cells, but not a lot is known about how it's happening within neurons. And due to the unique polarity and long-range transport that's required in neurons, there's a lot of basic questions just about mitochondrial quality control and dynamics that are not yet understood, such as are distal and somal mitochondria subject to the same quality control mechanisms? Where exactly is this mitophagy pathway occurring within this highly polarized cell? And also, how quickly are these damaged mitochondria being removed from the cell? And so today we're going to focus specifically on looking at the temporal dynamics of this mitophagy pathway. So here what I'm showing you is routinely what we can see if we damage mitochondria. These are within HeLa cells. What you get is the formation of these ring-like structures of parkin and optineurin that are recruited to this damaged mitochondria and start to engulf and encircle them. If you look at this within a 3D projection, it would be as a sphere surrounding that mitochondria. But here we're showing a single Z plane where it looks as if it's a circle. And so genetics links have suggested that failure to remove these damaged organelles leads to the buildup of mitochondria that could ultimately lead to cell death and degeneration over time, implying that the temporal dynamics of neuronal mitophagy may be critical. And so what we can do is we can use what we've previously known from HeLa cells, where we know the time course from damage to engulfment of that damaged mitochondria within an autophagosome is less than 1 hour. And so we wanted to know how conserved could this mechanism be within neurons. And so to address this question, we're using primary hippocampal neurons, where we're inducing mitochondrial damage via global oxidative stress through antioxidant removal, which we've showed here is AL3, or we're using low doses of antimycin A. And I'll note that we use these mild treatments to mimic highly metabolic cells. We then can ask, what are the spatial and temporal dynamics of these various mitophagy-associated proteins to these damaged mitochondria using live cell imaging, where we can monitor this in different cellular compartments. So in neurons, we visualized optineurin recruitment to mitochondria following an hour of antioxidant removal, looking at Z-stacks of the soma of a hippocampal neuron, where optineurin is shown here in yellow, and our mitochondrial targeted marker is shown here in magenta. And over time, what you'll see within this video is optineurin recruitment to these damaged mitochondria. So events within this Z-stack are highlighted with arrows, and I'll let it play just a couple more times so you guys can begin to see where those events are. We look down here at the bottom, you can see that they're occurring at different Z depths within the soma. And again, they look very similar to what I previously showed you in HeLa cells. Again, you have this optineurin ring that's engulfing or encircling this damaged organelle. And we confirmed that optineurin translocation is specific to damaged organelles using TMRE, which is a dye that measures mitochondrial health. So we routinely found optineurin rings that were positive for our transfected mitochondrial marker. However, they were negative for TMRE, showing that optineurin translocation is specific and that we can use this to positively identify mitochondria that are undergoing this mitophagy pathway. So using live cell imaging, we were interested in characterizing the temporal dynamics of neuronal mitophagy. You can imagine in healthy cells, this constitutive pathway is turning over damaged organelles, and this helps to maintain neuronal health. However, in a neuron that's expressing some of these disease-linked mutations of pathway components, this pathway becomes stalled, which would lead to the buildup of these damaged organelles, which could be detrimental to neuronal health and eventually cause degeneration. So to us, this suggested that the time course for the removal of damaged mitochondria may be critical. Sorry about that. Okay, so using optineurin using optionurine positive mitochondria as an identifier of these various mitophagic events, we then induced damage, and we just asked how quickly are these mitophagy-associated proteins being translocated to the damaged organelle. So within an hour of antioxidant removal, we observed the recruitment of parkin, ubiquitin, TBK1, as well as LC3. Again, they all co-localized with optionurine around these damaged or spherical mitochondria. And I'll note that the translocation of these proteins also occurred within the predicted time frame from what was previously characterized in HeLa cells. We then looked at the final stage of this pathway, which is lysosomal fusion and acidification, to fully degrade these damaged organelles. And that's when we got some really interesting data. So to observe the temporal dynamics of lysosomal fusion, we simply scored optionurine positive mitochondria as being LAMP1 positive or LAMP1 negative, where LAMP1 is a marker for lysosomes. First, looking in basal conditions or control conditions, we can see that most of these optionurine positive mitochondria are also positive for LAMP1, where if we looked in induced conditions, we see that only 50% of our optionurine positive mitochondria are LAMP1 positive, suggesting that mitophagy induction may stall lysosomal fusion, but it does appear as though the system can begin to recover. If we just look at our 1 hour treated conditions versus our 6 hour, we do see trends towards more LAMP1 positive. We then repeated these experiments, but instead looked at lysosome acidification using LysoTracker. And here we were really surprised to see that in either basal or induced conditions, that most of our events were LysoTracker negative, suggesting to us that lysosome acidification is slow. So to confirm these results, we utilized a dual LAMP1 reporter where we could simultaneously visualize lysosomal fusion as well as acidification. And so in non-acidified organelles using this reporter, they will be labeled with both the SEP as well as the RFP. But in acidic environments, this SEP signal will be quenched, leaving only the fluorescent signal of the RFP. We then again identified optionurine positive mitochondria, and we found events that were LAMP1 negative, suggesting that the lysosome had yet to fuse. We found events that were dual labeled with both the SEP as well as the RFP, indicating a non-acidified organelle. And finally, we found events that were RFP only, suggesting a degradative competent lysosome. We then repeated this with our experiments and scored this. And consistent with our previous findings, we observed that most of these mitophagic events in either basal or induced conditions were dual labeled and remained sequestered in a non-acidified organelle. So we further tested this model by performing SNAP pulse chase experiments where we could unambiguously label different mitochondrial populations. In these experiments, neurons are expressing a SNAP tag mitochondrial construct. We then perform our first SNAP pulse during the mitophagy induction. Cells are then washed to end the mitophagy induction and labeled with a SNAP block to saturate the remaining SNAP sites. And then cells are washed again and left for 24 hours. Mitochondria are then labeled with a second SNAP pulse prior to imaging. So this results in a spectrally distinct old mitochondrial population and young mitochondrial population where the network is dual labeled. We then looked at long-term sequestration to see whether our old mitochondria had been degraded. So 24 hours after initial damage, we observed that optionary positive mitochondria, and we found that that these mitophagic events were positive for the old mitochondrial marker. However, they were negative for the young mitochondrial population, indicating that these mitochondria had been sequestered for an extended period of time. These events had also yet to undergo lysosomal fusion, shown here by the absence of this LAMP1. Sorry about that. Okay, we then repeated these experiments that looked at acidification using LysoTracker. And again, we were able to observe events where our optionary positive mitochondria were only positive for the old mitochondrial population, and again, were negative for the young mitochondrial population. And these two had yet to undergo lysosomal acidification. So these events were observed in both basal as well as induced conditions, suggesting that there is this slow, inefficient turnover to degrade damaged organelles that occurs hours to even days after the initial mitochondrial insult. So what's really interesting about this is that this delay appears to be specific to neurons. We performed complementary experiments in HeLa cells where we looked at the time course of lysosomal recruitment following mitophagy induction. First, looking at lysosomal fusion, we determined that after 90 minutes of treatment, about 50% of our optionary positive mitochondria were also positive for LAMP1, and this increased with time after treatment. Again, looking at acidification using LysoTracker, we were able to see very similar things. 50% of our optionary positive mitochondria were also positive for LysoTracker, and again, this steadily increased with time. So it looks like in these non-neuronal HeLa cells that both lysosomal fusion and acidification are efficient following damage. So to conclude, our data indicates that one, we were able to establish a mitophagy induction paradigm where antioxidant removal induces these low levels of mitochondrial damage without compromising the network. And that again, we can use this optionary translocation and ring formation to identify mitophagic events. With data I wasn't able to show you, we've also been able to look spatially to see where within the cell that these mitophagic events are occurring. We think that they are preferentially positioned within the soma. So we think that there are other mitochondrial quality control mechanisms that could be potentially mediating some of the axonal mitochondrial population. And finally, today, what I was able to show you, looking at the temporal dynamics of neuronal mitophagy, we were able to see that autophagal engulfment is efficient. However, this lysosomal acidification may be a rate-limiting step in neuronal mitophagy, where these non-acidified organelles are persisting for many hours to even days after initial insult. And so I just want to bring you back to end with looking at these persisting organelles that are sitting there for days to even hours. And so whenever I give this talk, one of the things that always comes up is, why are these mitochondrial engulfed autophagosomes slow to degrade? So starting out my lab, we're really interested in following up on this question. Sorry about that. First, looking at what could be one of the possible reasons that there is a delay of degradation of these mitophagosomes. So one option could be that these mitophagosomes are actually failing to fuse with degradative competent lysosomes. Or another hypothesis could be that these mitophagosomes are fusing with degradative incompetent lysosomes. So with that, we're trying to separate these two by looking first at measuring the local pH and protease activity of some of these mitophagosomes. To do that, we're using a cathepsin D reporter where again, we can monitor whether or not these are acidified by being either dual labeled or single labeled. We're also starting to characterize the ultrastructure of these non-acidified mitophagosomes using CLIM. So with that, I will acknowledge my my postdoc lab, Erica Holzbauer at the University of Pennsylvania, and all the members of her laboratory, as well as starting out my lab. We have 2 members thus far. Also want to point out that we're hiring, so if any of this interests you, please follow up with me. And with that, I would be happy to answer any questions. Thank you.
Great, thank you, Chantel. We now have time for a few questions and that are coming through the Q&A. Irina is asking, could you please comment more on how you removed antioxidants from the system to create an antioxidant-free environment for your cells?
Yes. So one of the components of media that are necessary for neurons is B27. And so there is a B27 that is antioxidant-free. So with that, there are 5 essential antioxidants that are removed from the media. Those are, let me see if I can find them for you. Those are vitamin E acetate, superoxide dismutase, catalase, and glutathione.
Okay, thank you. And there are more questions for you in Q&A. If you can type them, that will be great.
All right, thank you.
Thank you. Okay, our next presenter is Dr. Brian Balliveau. Dr. Balliveau is an assistant professor of genome sciences at the University of Washington. His lab uses computational and molecular tools in combination with super resolution and advanced quantitative microscopy to investigate the causes and consequences of 3D genome organization. Today, he will be giving a talk titled Oligo-Based Technologies for Programmed Spatial Biology. Welcome, Brian.
Thank you very much, Gökhan, for the introduction, and thanks to the organizers for the opportunity to speak. So in my talk today, I'm going to highlight some technologies that our lab has been involved in developing, and kind of the theme here is we're trying to look at what the cutting edge is in terms of spatial tools for looking at things like genome organization or gene expression in individual cells, and to try to make those more broadly available to the community to harness in their own experiments. So with that, I'll jump in. Okay, so our lab is an imaging lab. We image lots of things. My favorite thing, which I'm going to highlight here quickly, are chromosomes. So we have a lot of interest in the genome and its organization. An easy way to look at genome organization is when the sample kind of does it for you. So if we have spread chromosomes like this, it's really easy to look at these and have a sense for what's going on with this genome, say in terms of the number of chromosomes or their size. But when we look most of the time in asynchronous cells, at least, and staying with the DNA dye, we see something that looks really different. So we see this kind of splotchy, smeary mess of blue signal. So the central biological challenge that drives a lot of our technology development is trying to build tools that allow us to ask questions about how this genome is structured and how that influences its biology. And kind of the 30,000-foot view of why we and many other people are interested in this is trying to understand at some level how 2 linear meters of DNA can fit into a compartment that's just a few microns in diameter, contain all of the genes in our genome, and tens of thousands or more of regulatory elements that can help potentiate different transcriptional programs. So we know there's a big spatial component to how that biology plays out, as well as biology related to DNA repair and DNA replication, to name just a few processes. And there's a lot to kind of poke at in terms of having single cell imaging tools to try to investigate those phenomena. So our lab has a strong focus in developing tools for looking at these kind of things. I'm going to introduce a few technologies as I go in this relatively short talk. So the first one is Oligopoints. This has been around for a bit now, but it's an important platform that the other technologies are going to rest upon. So I'll tell you a bit about these programmable fish probes and what we can do with them. Once we have these probes in hand, we can do a couple of fun things with them. One that I'm not going to go into today, but something we like a lot is using highly advanced microscopy, single molecule localization microscopy specifically, to try to zoom in on what the structure of chromosomes look like on a nanoscale. And something I will highlight today is a technology that has been introduced relatively recently called SABRE, which is a new way for doing multiplex signal amplification in cells and tissue. So to jump into Oligopoints first, this is a technology that I co-developed with Eric Joyce when I was a grad student in Ting Wu's lab quite some time ago now. And the basic idea is that instead of doing the FISH or fluorescence in situ hybridization approach, using DNA that we isolate from the host's genome, meaning genomic DNA or cDNA for Rna Fish, we start with all libraries of synthetic DNA or oligos that we can design computationally and program the components of. Because these typically come in terms of the bang for the buck in a nice format, in terms of we can get a lot of oligos in terms of the quantity themselves for relatively cheap, all things considered, but the amount of material ends up being pretty small. We put that through some molecular biology to amplify the material and convert it back into single-stranded DNA so it works well for fish. And that allows us to go through this whole process. So the upshot of this is that we have a set of protocols that we've developed that allow us to do this computational design, target things that are very small for DNA fish on the order of tens of kilobases or smaller, or things up to the scale of megabases, essentially with the same protocol for this process and the same protocol for the fish. And it's robust, scalable design to do it, which is great. I think that the key interest we have in oligopaint type technology or oligoprobes is our ability to engineer all aspects of them. So we have full control over the sequence of everything that we order, and that allows us to have fun with the design. So kind of the base unit of one of these probes would be a segment that targets it to genomic DNA and or an RNA. And often because of the amplification we do, it'll have a priming sequence. We can use that priming sequence to add a fluorophore that makes it easy to see and straightforward to label a whole bunch of different sequences. Alternatively, we can just program in overhangs, or we can call them binding sites for secondary oligos that would be analogous to secondary antibodies binding primary antibodies. And this provides a lot of flexibility in experiments. So by being able to engineer this whole substrate, we can focus in on different regions and empower different kinds of new fish technologies. One example would be allele-specific fish by honing in the targeting to land on sites of phased and known single nucleotide differences. And other examples that are more relevant for the rest of the talk are taking advantage of these sites that we can encode and selectively visualize at different times in the experiment using iterative hybridization, where we hybridize some but not all, image them, remove the signal in some way and add more. And nice examples of that are now termed spatial genomics. So here's an example from Jiawei Zhang's lab where they target 34 distinct sites on the human chromosome 21 to view its folding conformation in 3D space. And of course, the really popular spatial transcriptomic technologies that use imaging, such as the MERFISH from the Zhang lab and SeqFish from Lan Kai's lab. So I'm going to tell you about a technology next called SABRE, and this is something that came during my postdoc time. So this was a really fun project with a lot of people involved that I want to give credit to first before I jump in. So this project was led by Josie Kishi, Sylvan, Emma, and myself in the labs of Pengyan and Connie Supko. And the start of SABRE is another acronym in technology, which itself is not essential to understand for what I'm going to show in cells, but I think this is a really nice and elegant method that Josie Kishi and colleagues introduced, and it is essential for how we get the tool we need. So this is a technology where you can have priming sequences, and these are essentially pieces of DNA, single-stranded DNA that have a 3' hydroxyl and can be extended by a polymerase. So very similar to a PCR primer. And we put these together with hairpins that can act catalytically to enable extension and strand displacing polymerase. So I'm going to go through the reaction cycle relatively quickly, and then there's a video coming that might make it more clear, and happy to answer questions after that. So the basic idea here is that we have a priming sequence A. In this scheme, its reverse complement would be A star, and we program the hairpin to be complementary to the primer. So they can sit down by base pairing, and then in the presence of a strand displacing polymerase, we can get extension all the way up into this little black double dot bar here. This is a key component of the system. This is a stop sequence. It's a way we can stop the polymerase from synthesizing further. There's a few different tricks you can use to make this happen. And if you're able to engineer this situation, then you end up in a state where you get a branch migration equilibrium between the state where the B on the hairpin is pushed back and the state where the B on the newly synthesized AB concatemer is pushed back. And the final element of the design that's important here is that the A star interaction is designed to be relatively weak and transient, such that spontaneous dissociation will basically pop this off. So going through one cycle, what we've done is taken A and added the B sequence onto it. And since the hairpin itself was not modified in the reaction, it's free to go on and catalyze additional extensions on primers that haven't been extended yet. Okay, so where this is going to get into the cellular space is what happens if you make the special case where the sequence that we're adding via the hairpin is the same as the priming sequence. So then instead of a one-step addition and done, this reaction can just go on and on and on as long as you let it go. And this is a really simple and straightforward reaction. It's isothermal. It runs at 37. You can get hundreds of bases, thousands of bases even of extension depending on the conditions in just a few hours. Really simple and cheap to pipette. So it's something that we were looking at when I was in this lab at the time and thinking, wow, this is a really powerful way to make some really useful DNA, because repetitive sequences like this make great scaffolds for signal amplification. Here is a movie showing the process I just described, somewhat fast. Primer binds, extension pops off at the stop sequence, and this can just happen over and over and over when you have the same sequence being copied onto the primer. So how this comes back into the cellular context is that we have this approach SABRE, or signal amplification by exchange reaction. So this comes from the PER reaction. And kind of the joke here is that this is like a lightsaber. So we're taking something like a fish probe or something that can dock at where fish probes are hybridized, and we're extending it using this in vitro reaction that places concatenamers of ssDNA on it to create scaffolds to make the probe long and bright, just like a lightsaber would be. In practice, what we're really doing is adding somewhere on the order of several hundred bases or more of ssDNA onto our probes. And we use a fluorescent labeled complement, reverse complement that is, of 2 copies of the binding of the sequence we polymerize, which is a 10-mer. So it ends up being a 20-mer oligo. So we can create dozens of binding sites for this fluorescent oligo and amplify our signal somewhere on the order of 10 to 40-fold over baseline. So SABRE is relatively straightforward to conduct. You do the PER reaction in vitro. You do your in situ hybridization like normal. You can even use the extended oligo probes if you want, or you can use normal oligo probes that project a binding site like I described earlier in the talk, and then hybridize in a later step the reverse complement that's been extended by PER to dock the concatenamer that way. And then at the end, you just add your fluorescent oligos in 1/3 additional but quicker step to allow visualization of the scaffolds that you've placed here. So I'm going to show you a little bit of data now, just validating that adding SABRE on top of fish doesn't change the behavior of fish, which is an important thing for us to check. So we validated for DNA fish that the assay is working as expected. Back on my favorite system, the metaphase chromosomes, we placed some spots adjacent to one another on human chromosome 1, looked at metaphase spreads and saw the colocalized pattern that we'd expect. And we also see that in interface cells. For RNA fish, the standard's a little bit different. So typically what we do is we take a transcript, we look at the fish probes that bind along it, we split them into 2 pools. So each one of these is polymerized with each of these pools. So there's a green pool and a magenta pool are polymerized with a different hairpin sequence. So they have a distinct orthogonal sequence of concatenamer on them. And then we hybridize in 2 channels and look at the concordance between them. And after automated image processing, you can see that the concordance between the 2 channels is excellent, very, very high for an assay like this. So we're convinced from these experiments and others I'm not showing that RNA and DNA SABRE fish aren't inherently changing the underlying assays, but they are giving us a way to make brighter signal in a relatively easy and straightforward way. I think the real power for this kind of approach for us comes from its ability to multiplex. The amplification is certainly nice. It always helps to have more photons when you're imaging. But the multiplexing is really what's starting to push the boundaries, I think, of what's possible for these kind of approaches. So I'm going to take you through how we do this. Here's an example from Sylvan Lapin and Connie Sapko's lab. We did this fish experiment. So what Sylvan did is he hybridized 7 different RNA targets with probe sets. Each probe set has a distinct orthogonal concatenamer that's been added to it by PER. We have 50 of these to choose from. Sylvan's chosen 7 distinct ones here. And there's also 2 protein targets we're going to use conventional immunofluorescence to visualize in the last round. So in the first round, after hybridizing all 7 RNA probe sets at once, we only add the fluorescent oligos that are complementary to the first 3 targets in 3 distinct channels and image them. And we get an image that looks like this. This is a mouse retinal tissue. So the key thing that makes this work is that we are able to engineer the thermodynamics of the fluorescently labeled oligo that binds the concatenamer to be stable under these conditions, but much weaker than the thermodynamics that hold the fish probe onto its reverse complement. That allows us to change the solution conditions by adding chemical denaturants and to displace the fluorescent strands without disrupting the binding of the fish probes to the RNA targets, or if we're doing DNA stuff, the DNA targets. That allows us to then reuse the same fluorescent channels with distinct concatemer sequences on 3 distinct targets, and in theory, iterate this many times. So we just did it 3 times here on 7 targets plus 2 conventional immunostaining targets. But this could be done many times. And we have other data I'm not showing where we've done this 5, 6, 7 times or more. So the upshot here is that we have a nice technology with amplification in tissues that allows us to, for instance, look at cell site specific RNA expression patterns, where here in Connie's lab, they know the biology of the retina really well. So they were able to kind of pick markers that would give us beautiful staining patterns like this. But in terms of the overall application space, where we see things like this going is having things like single cell RNA sequencing data sets, which we've already seen kind of today, and have it going into the dimensionality reduced space and trying to identify marker genes that might help us visualize rare cell types or cell states, and then to use techniques like Saberfish to look at the spatial arrangement of these in the tissue that the data set came from. I'll also mention very quickly that this is not restricted to DNA and RNA. So Sinan Saka and Yu Wong in the lab also showed that you can take antibodies and conjugate them with oligos and then polymerize the reverse complements of these using PER. And this allows you to do immunosaber. And here showing a 10-plex assay plus DNA staining in mouse retinal tissue. So if you're interested in Saber, the developers have really worked hard to try to make this accessible. There's a saber.fish website that has a lot of information about how to do this technique collected. There's also an email list that we maintain where we field questions from people trying to use the technique. So we'd really like to go see this out in the world doing awesome stuff. So if you're interested, this is a great place to start. I'm going to close just by highlighting a couple other quick things about that we've introduced more recently to try to help researchers get off the ground with technologies like this. So the first one is how to design the probe sets that would encode what you would need to do for an RNA Saberfish experiment or a DNA Saberfish experiment, especially if it were multiplexed. And this is via a tool that we've introduced called PaintShop. So there's already nice informatics support for designing fish probes on the genome-wide scale, but even for labs like ours that do a lot of fish and do a lot of informatics, it's not always straightforward to target many different things at once, keeping track of what you're encoding, say with different Saber concatenamers or different bridging oligos, secondary oligos, et cetera. So that need to simplify this and make it more accessible really drove the development of PaintShop. And this is work that was led by Elliot Hirschberg when he was a technician in the lab, and Connor Campbellson, who's still a PhD student in our lab. So what PaintShop does is it provides a web framework to simplify a lot of the common tasks you'd have to do in the kind of process like this, like for instance, going from a genome scale collection of oligo probes and mapping them onto specific transcripts and making sure the polarity of the oligo probes are correct to bind the sense or antisense strand, depending on what you need to look at. And we also have utilities for changing the number of probes you order just to save money or to normalize them across targets. And the big one, appending functional sequences like the sequences you would need for PER or to amplify them in the molecular biology pipelines I mentioned earlier in the talk. So PaintShop is a great place to kind of orchestrate all of these common tasks. We have pre-computed the transcriptome sets for a lot of different organisms. You can do batch retrieval using RefSeq IDs as your queries against many targets in parallel. And we also have the ability to do this for DNA and or for RNAs that aren't RefSeq. You can put in bed type information and get these out in batch the same way. We currently host about 300 million different oligo probe sequences across 9 model organisms and 13 genome assemblies. And we're planning to try to continue to update this as we go. And one nice aspect of PaintShop that we've added in terms of thinking about the probe design is that we have a machine learning model that's able to get a very accurate prediction of how specific or not any given probe would be. So that is a way users can decide which probes they want to order or not as they consider how to trim or balance their sets. So this is all available at paintshop.io. There's a few GitHub repos that support this as well. This is now published and it's up and going. And if you want to check out the paper, we do have more metaphases, of course. So this is just my last slide. I also want to give a shout out to Lily Deng, who's a staff engineer in the lab. So Lily's been working on the development of an automated fluidic system. This is something you can buy currently, but Lily's system is a fraction of the price of what a commercial system would cost, and is really feature driven around doing things like multiplexed RNA fish and Saberfish. So this is something that's going to be on bioRxiv or archive probably in the next month or so. And if you're interested in getting going with fluidics in your lab, this is something you can, we hope, get set up with for just a couple $1,000s. And it has a lot of features that make it well adapted for things like saberfish experiments involving exchange. So with that, I'd like to thank the organizers again for the chance to talk, and I'm happy to take questions.
Thank you, Brian. Beautiful images and technique. There is actually questions in the Q&A, and Ru is asking, how does your method compares to merfish and seekfish? Is it the amplification only?
Yeah, so I'd say saberfish is a way to do fish, and merfish and seekfish are ways to encode information into multiplex fish experiments, if that makes sense. So you could use saberfish as a component of a merfish experiment, and some people are in fact doing that. There are instances that are published, nice instances where people have either kind of done a saber-S thing without using PER, just by buying long oligos that are pre-extended from the Zhang lab. And then some of the seekfish work has used a hybridization chain reaction as a way of doing amplification. So that's related in some ways to the PER, but I think the big difference is that that's an in situ amplification versus amplifying something in vitro first. And there's probably pros and cons to both approaches, but if they're working well, at least the output is similar.
Thank you. We are going to take a brief break right now, 5 minutes. We will plan to return around 9:56 with the next set of presentations. While we are on a break, I would like to remind you again that this is day 1 of 2 of the symposium. We will be hosting day 2 from 8:30 Pacific time to noon again tomorrow. Thank you and see you soon.
All right. Hello, everyone, and welcome back from the break. My name is Rithvik Basan, and I'm a scientist on the modeling team at the Allen Institute for Cell Science and a member of the Symposium Planning Committee. I'll be the MC for the remainder of the day. The next presentation comes from my Institute teammates on the EMT team. Here, EMT stands for epithelial to mesenchymal transition. They will be talking about using the EMT transition in HIPSCs as a model to study cell states and state transitions by combining cell behavior, cell organization, and cell identity. So today, Antoine is going to kick off the talk for the team. Welcome, Antoine.
Thank you. Hello, so I'm Antoine, a scientist in the Allen Institute for Cell Science, and we are a very large team working exactly on using the epithelial to mesenchymal transition in human-induced pluripotent stem cell as a model to study cell state transition by combining cell behavior, cell organization, and cell identity. As most of you know, as most of you know, the EMT is an epithelial to mesenchymal transition, and during which the epithelial cell will lose their epithelial characteristic. They will we will show the cytoskeleton will be remodeled, they will lose the apical basal polarity, and their cell-cell adhesion will be weakened. By the end of the classical process, the cell will be individualized motile mesenchymal cell. The EMT occurs in numerous different contexts. It has been first discovered in a developmental context, but it's also known to be part of wound healing and play a big role in cancer metastasis. Because of all this context and the plasticity of the process, a clear definition of the EMT is difficult. And it's been highlighted by a recent paper on the behalf of the EMT International Association, calling for better and more comprehensive definition of the EMT. And this is the challenge we choose to tackle. Understanding better the EMT provides us actually a great opportunity to study cell states and state transition. On the right part of your screen, you can see schematic of how cell state and cell state transition could happen during the EMT. There could be either one or several starting an epithelial cell state and ending mesenchymal cell state. And cell could use either all the same paths and go through the same cell state to go from E to M, with possibly a different duration, each cell staying in a different time in all the cell states, or there could be multiple paths to go from E to M. Finally, it's possible that each cell create its own path through the cell state to go from E to M. So how are we going to address this question? How are we going to define the cell state during the EMT? Well, we're going to show you today that we are planning to integrate data from multiple data sets. And you'll see time lapses, single cell RNA-seq, Rna Fish, and immunolabeling. The goal is by all these data sets and by looking at the cell from all these different angles, we can define cell behavior, cell identity, and cell organization. And with all this information, finally define cell state. So how are we going to do that? In which model? Well, we're going to use the differentiation of human-induced pluripotent stem cell into cardiomyocytes. The first step of this differentiation constitutes an EMT that I'm going to show you now. So here on the main part of your screen, you will see a movie that I'm going to play soon, where you will see a colony labeled for the plasma membrane going through the EMT-like process that we're inducing in those cells through the activation of Wnt signaling by the intermediate of an inhibition of GSK3 beta. So this movie is taken from 0 to 48 hours following the start of the EMT-like process. and an image is acquired every 20 minutes. So I'm going to play this movie, and what you can see is 3 different big main phenotypes. First, you see at the start of the process, the cell and the colony compact. And in the second time that we're seeing now, around the 24-hour time mark, cells start to migrate, isolate and migrate away from the colony. Finally, you can observe cell death and cell division throughout the process. So the first phenotype, as you're seeing again right now, is this compaction of the colony and the cell. And we wonder what was the role of the contractile apparatus of the cell in this cell and colony compaction. So to explore this, we performed very similar time lapses that I'm going to show here, but the only difference here is the cell line used, which is actually labeled for the actomyosin bundle through a labeling of the non-muscle myosin 2B. You will be able to see here 2 different movies, one on the left center on a small colony, one on the right of your screen center on large colonies. And before I start playing this movie, here you're seeing the image at T0, and what you can appreciate is the specific localization of the actomyosin bundle at the periphery of all the cells in the colonies at the start. And as I'm playing the movie, you can appreciate that the cell at the periphery of the colony rapidly lose this specific localization of actomyosin bundles, whereas some cells towards the center of the colony retain it. So first we observed early on an heterogeneity within our cell population, with cells retaining the specific peripheral localization of actomyosin bundle and some cells that have lost it. Another interesting thing is that the cells that actually retain late the specific localization of actomyosin are not randomly distributed throughout the colonies. Rather, they cluster together. And this clustering, these islands of cells, we wondered if this was due to cell-cell junction. And so we looked and we performed similar experiment, this time for a cell line labeling the tight junction, label for the tight junction with a label specifically of 0,1. You can see here again 2 time lapses, one on the center on a small colony, one on a large colony, the large one on the right part of your screen. And again, we see a similar pattern here. At the start of the process, all the cells show a nice localization at the periphery of the tight junction. And when I play the movie, you can see and appreciate that the cell at the periphery of the colony are the first one to lose the specific localization of 0,1. And we have clustered of cells retaining the specific 0,1 localization late in the process. So we wonder if the cluster of 0,1 and myosin were the same and if one controls the other. But we don't have a cell line label for both structures at the same time. So we turn toward immunostaining. And what you can see here on the left part of your screen is an image of a colony labeled for 0,1 in green, fixed at 24 hours and stained against myosin 2B in magenta. What I want to draw your attention on is that if you look at the cluster of cells showing the peripheral localization of 0,1, you can appreciate that there is 2 types of clusters. There is large clusters, and there is what we call narrow 0,1 strings. So I zoomed on the narrow 0,1 string in the central column here and on the large cluster in the column on the right. What we see is that in the large 0,1 cluster, we observe still the peripheral localization of actomyosin bundle. But in the narrow 0,1 string, this specific localization is lost. So it's very tempting here, especially knowing that the specific localization is present in all the cells at the start of the process. It's very tempting to think that we identified here a sequence of events of the EMT where all the cells have the peripheral localization of myosin and 0,1, then it's lost, the myosin localization is lost, and finally the cell narrow and you see their apical area getting smaller, and finally the 0-1 specific localization is lost. But to actually draw this conclusion, we would need much more time visualization of this process. And we're actually interested in understanding the EMT sequence of events. And rather than performing similar experiments on numerous different markers, we decided to go large scale and starting to turn to MNSX throughout the EMT process. And Stephanie will introduce that process.
Thank you, Antoine. Hello, my name is Stephanie Din. I am a research associate here at the Institute. And as Antoine said, we performed a single-cell RNA-seq experiment so that we could get transcriptional data to better understand the sequence of events in our EMT-like system. This experiment encompassed the 48 hours after the onset of the Wnt signaling pathway. This includes our initial induced pluripotent stem cell state at 0 hours, and then the progression towards a mesodermal mesenchymal state in 3-hour increments. So here is the data from that experiment visualized in a UMAP plot. This is just a way to visualize high-dimensional data in a more digestible 2D way. Each dot on the plot represents a cell. Its location in proximity to the other cells has to do with how similar or different it is in transcriptional profile. Here we have the cells color-coded by the time point in which they were collected. And you can see that all of our time points nicely cluster together sequentially across the plot. Where we see our 0-hour time point is in red on this right.
I think is Stephanie frozen.
Yeah, Sarah or Antoine, is one of you want?
Yes, we are happy to continue for Stephanie. So what she was showing here is that the UMAP of the transcriptional profile of the cell, oh, this kind of moved, but it's okay, show the nice evolution and the continuity of the evolution of the transcriptional profile over time. If we move now to looking more at, I don't have a control of the slide. Thank you. So here is the expression of different transcript during the EMT. What you're looking at, it's a selection of genes, and I'm gonna explain why those are selected in a little while. But for each of them, you see the evolution over time, and you see different bonds. The size of the bond, the circle, indicates the percent of cell expressing this transcript, and the intensity of the blue color.
Antoine, I think Stephanie's back if she wants to continue.
Excellent, Stephanie, if you want to continue.
Thanks, Antoine, for jumping in there. Somehow my internet cut out exactly when it was my turn. What Antoine was saying is that, yeah, the intensity of the color represents the level of expression for that gene at that corresponding time point. So if we look at the bottom set of genes, this list of genes is comprised of stemness and epithelial markers. And if we look at the plot, we can see that it is high and abundant in expression at the beginning of the time course, but then decreases as we proceed to the 48 hours. In contrast, if we look at the upper set of genes, we can see that these are comprised of mesenchymal and mesodermal markers, and we see the opposite trend, where the genes are low in expression and abundance, but increase by the 48 hours time point. If we look at this whole thing overall, hopefully you can appreciate that we can see what appears to be an inflection point in the middle of the time course, where there is a drop in stemness epithelial markers, but a notable rise in the mesenchymal messenger markers. This 20 to 30 hour time point may be familiar, hopefully. As a reminder, I will bring up our time lapse that we showed earlier, because this time point actually corresponds to a time frame of interesting cell behavior. Specifically, if we see starting now, the onset and growth of migration activity. So this shows how we can draw similarities between our transcriptional changes to resulting cell behavior. But now my colleague Sarah will show how we actually try to connect these two data sets by adding spatial information using FISH and immunolabeling.
Thank you, Stephanie. I'm Sarah Carlson, and I'm a research associate in the assay development team in cell science here at the Allen Institute. So I'll be taking you through the next couple of data sets. So as Stephanie showed you, it's interesting and informative to know from the single-cell RNA-seq data that we have different populations of cells that are expressing different markers within the same time point. So wouldn't it be nice to know whether these populations are somehow connected to a cell's placement in a colony? You know, we can't get that information from single-cell RNA-seq alone. So that's where Rna Fish and immunofluorescence can help us. So Rna Fish gives us transcript localization. Here I'm showing you Wnt5a and DKK1, which are genes involved in the Wnt signaling pathway, which is what we are targeting in this system to induce CMT. This image shows fixed cells at 27 hours post GSK3 beta inhibition. The 27-hour time point is one that I'll be focusing on a lot in this presentation because we have, individual cells starting to migrate while also maintaining that compacted centers of the colonies, which is good for showing multiple cell behaviors. So here we see different spatial distribution for both genes. So higher DKK1 signal sort of near these compact centers of the colony, and higher Wnt5a signal in cells near the edge in green. There's a large amount of heterogeneity within these populations. And so if we go further into that direction of spatial patterning at the colony level, we can also look at protein expression in our immunofluorescence data, where we see that some proteins also have a spatial pattern of expression depending on a cell's location at the colony level. Most obviously cells within the compact center of the colony, versus at the edge versus cells that have already migrated from the colony. So you can see that in this example of cells fixed at 27 hours post GSK3 beta inhibition, they're labeled with EOMES and brachyuria antibodies, which are mesodermal transcription factors. Generally, we see higher levels of EOMES expression, like I said, in the center packed region of the colony, and decreased levels at the periphery. And we see the opposite in brachyuria expression. So there's, more EOMES green here in the center and pink brachyuria on the outside. If you notice this white box near the edge of the colony, if I zoom in on that boxed region from the previous image, we can see that even within this region, there's a bit of variability. So the arrows near the top of this region are pointing out cells with high brachyuria and low EOMES signals, so more pink than green. And you can compare those cells to the cells being pointed out near the bottom, which points to a cell with high EOMES and low brachyuria signals, so more green than pink. And then yet again, compare those cells to the cells indicated here by these arrows, which show sort of low expression for both of these markers. And all of these cells are near the edge of the colony, but show, differing signal intensities for these markers. So could this variation indicate different cell state paths? We have separate sets of data that give us different snapshots of information, right? Suggesting that cells undergoing the CMT are different from each other, but we aren't getting information about the path they took to get there. So what were they doing in the time leading up to these fixations? Where were they going? And can we connect fixation and staining to time lapse? We think that by watching a time lapse followed by fixation and staining with markers of interest, we could try and link cell behavior to protein expression. So this is a time lapse of our alpha-tubulin MEGFP line, where images were acquired every minute for 2 hours, about 29 hours post GSK3 beta inhibition. We see cells being very dynamic this time, right? So they're migrating away from the colony, they're touching each other in the open space and moving away from each other again. They're going, really far in one direction or maybe not moving at all. And don't forget dividing cells, which I think this guy divides right here in the center. So in the next slide, I'm going to show you post time-lapse fixation. So I'm showing you the EOMES brachyuria pairing from before, and I'll repeat that by watching that time lapse followed by fixation and staining with markers of interest, we could try and link migratory cell behavior to protein expression. So do we see patterns of perhaps different kinds of migrating cells, depending on what direction they're migrating or how they're moving or what cells they're interacting with? So a lot of the migrating cells in this image, are perhaps maybe similar in brachyury expression. So we see a lot of pink, but there are a couple cells with, comparable EOMAS expression shown in green, but it's hard to tell at this magnification. So what about cell movement is influencing expression of these proteins? And then I'm going to show you an example of a higher magnification time lapse followed by fixation. When I play this video, you'll see the cells in this field of view have different behaviors and movements. So try to just look at the whole field of view at once. And the video will end with the fixation and staining. And you can see that these cells also seem to be showing different patterns of expression. And we're working on trying to connect cell motility and behavior with their patterns of expression after fixation. So Mackenzie is going to take you through a related element of integration that we are interested in, which is focused more on time lapse.
Thank you, Sarah. Hi, everybody. I'm Mackenzie Costin. I'm a research associate here at the Allen Institute for Cell Science. Like my colleagues have mentioned, we're integrating multiple different types of data to better understand how EMT cells act over time, and also to understand the root of variation of different marker expressions that we're seeing. We can also take a new and different approach to data integration to explore the EMT cells, using an approach that we've used in the past to study the human-induced pluripotent stem cells. So previously, we have used high throughput 3D imaging to image multiple different cell lines with unique different structures tagged to integrate the data together and create a holistic integrated view of the HIPSC. Within this model, we can see the organization of the HIPSC and also see how unique structures are interacting together. In order to do this with our EMT cells, though, we needed to take a bit of a different approach. The first thing we did differently was to decide on a distinct time frame to study the EMT-like cell. We decided to look at the early migrating EMT-like cell. So we imaged cells approximately 25 to 30 hours after the GSK3 beta inhibition begins. In order to accommodate this in our imaging pipeline, we need to account for time. So we image cells for 20 minutes during this time period in 3-minute intervals. And since we're imaging these cells at high magnification in 3D, we want to be cognizant of cell health by exposing them to not too much laser. So we image at every 3-minute interval with transmitted light, and for every other 3-minute interval, we use laser so that we still get all aspects of our imaging. This is an example of what this data looks like. And unfortunately, it is not playing for me, which is very sad. We troubleshooted this beforehand, but I will describe it in detail. So this is the lamin B1 cell line. It's showing the nuclear envelope. This cell here would have been not moving around very much, except on the periphery, a little bit of movement. This upper cell here would have been moving up here, and then it would have come back down, even touching another cell and then moving back out on its own. So this highlights the heterogeneity that we see of this early EMT cell migration. Lots of different movement behaviors are observed, which is really neat. And we can also see here, where we would be seeing, we could see cell behavior in terms of migration, but also we could see how the unique structure within the cell moves along with the cell motility. So as we go forward with this, we want to image multiple cell lines, each with distinct and relevant cell structures tagged. For example, we've looked at the Golgi, actomyosin bundles, plasma membrane, and the nuclear envelope. We can then integrate all these different data types together to get a holistic and integrated view of the early migrating EMT-like cell with respect to its movement behavior, with respect to the individual structures, and we can also see how these structures change as the cell moves. So with that, I will pass it back off to Antoine, and he can conclude our presentation.
Thank you. So today, we hope we showed you our goal and our intent to integrate data in two different ways: integrate data across data set and integrate data from the same data set in the similar fashion that what we performed recently on the HIPSC integrated cell, but here to create an integrated EMT cell. And the goal is looking at the cell from all this angle once again to recover, my screen doesn't move, recover the cell behavior, cell organization, and cell identity of the cell to define properly the cell state and to address the question of which cell state can we identify during the EMT and how cells change their state during this process and how the cell transition occurs. And we really hope that next year we can tell you more about all this exciting science. In the meantime, I would like to thank absolutely everyone at the Allen Institute, because if we were just a handful speaking today, absolutely everyone is involved in this project. And we wish to thank the Allen Institute for Cell Science founder, Paul Allen, for his vision, encouragement, and support. Thank you.
Okay, thank you, Antoine, Stephanie, Sarah, and Mackenzie for a great talk and for dealing with internet issues. We now have time for a few questions that have come in through the Q&A. Austin asks, any idea why there is a divergence at the beginning time points of the UMAP? Could these be the cells that first initiate the EMT process?
Stephanie, I think this one is for you.
Yeah, totally. Yeah, that region of the UMAP was also of great interest to us, especially that division between the 0 hour and then the onset of differentiation. We're still digging into this because this is fairly new data, but your hypothesis is similar to ours. Since the 0 hour is the induced pluripotent stem cell state, I don't think it's unreasonable to think that after the onset of differentiation, there's some drastic changes. So.
Okay, and another question from Timothy. Do these changes depend on cell movement? If the cells could not crawl away, would the Iomes brachyury shift still occur?
Well, this is actually something we're exploring. We're not ready to answer yet, but it's exactly the goal of integrating data from different data types. What I mean is that looking just at snapshot of Iomes brachyury is interesting, but we gain much more information at looking at that with adding the motility, and we can retrace at least some part of the cell history and its motility to define better cell state.
Okay, great. Thank you. There are a few more questions in the Q&A, and if you could answer them, that'd be great. Thank you again, EMT team. Our next presenter is Dr. Joe Schoeneberg. He is an assistant professor in the departments of pharmacology and chemistry and biochemistry at the University of California, San Diego. His research group investigates the fundamentals of mitochondrial dynamics and the role of mitochondria in neurological diseases in 4 dimensions, with the goal to develop a new generation of drugs based on mitochondria. Today, his talk is titled Integration of Particle Simulations, Lattice Light Sheet Imaging, and Deep Learning in Quantitative Cell-by-Cell Biology. Please welcome Joe.
Hi, thank you, Ritwik, for that fantastic introduction. Hello, everybody. Yeah, my name is Joe Schoeneberg. I'm really excited to be here today, and I'm really excited to be talking about in this data integration session today, because we are integrating particle simulations, lattice light sheet imaging, and deep learning to do quantitative cell biology. And so in my lab, we essentially have these 3 sections. So on the left-hand side, I depicted the bottom-up approach, where we essentially take system parameters and particle-based simulations like this READY framework that we developed to then create particle coordinates. And from these particle coordinates, you can essentially convolve them with point spread functions from the microscope, noise from the microscope, to arrive at simulated synthetic microscopy images. And on the right-hand side, I have the top-down approach, which is essentially where we use adaptive optics lattice light sheet microscopy to directly investigate cells, live cells in organoids. And then we essentially develop these segmentation tools, tracking tools, to then arrive at coordinates and system parameters to really understand the system. And our deep learning that we use in the lab, that is essentially to tie those two columns together. So let me walk you through how we are doing this in the lab. My talk has essentially three parts: the bottom-up part, the machine learning part, and the imaging part. And then I'm going to talk a little bit about what Ritwik essentially hinted at the mitochondria that the lab is focusing on right now. So, but for this section, for like essentially giving the broader overview of our data integration, I'm going to talk not about mitochondria, but about this system that's called Clathrometered Endocytosis. So Clathrometered Endocytosis is incredibly important because that's the process where the cell takes up metabolites, proteins, hormones, receptors, and also COVID-19 from the outside of the cell into the inside of the cell. And the key proteins here to watch are called clathrin. Those are the molecules that form these cages, and dynamin, which are these little molecular scissors that cut the necks off of these vesicles once they are mature and can be split off the plasma membrane. And so what we have been faced with when we wanted to study that system from a simulation point of view, that there was this gap in particle-based cellular simulation tools. So on one hand, you had like molecular dynamics packages, but they were lacking reactions. On the other hand, you had these cellular simulation packages, but they were lacking crowding. And so that is essentially what I decided for my PhD to build a new particle-based reaction diffusion engine. And that we call that READY for reaction diffusion dynamics. And it's essentially an overdamped Langevin integrator plus reactions. And so it's a big tool, hundreds of thousands of lines of code, C++ with GPU acceleration. It's open source. You can check it out on GitHub. And what you can do with this is you can, essentially create these little toy simulations. This is a predator-prey toy simulation where you have these nutrient generators, and then you have like these predators that are like eating up the nutrients, get bigger and then split. But that's just like a toy one. We use this for multiple systems. We studied synaptic vesicle release. What is extremely exciting now, the Allen Institute is using READY as an engine for the virtual cell. And what I'm showing here is Blair's work, where she uses READY to essentially visualize actin polymerization and microtubule dynamics in the cell. So that's super exciting. I'm not going to talk about this. I hope we're going to hear a little bit more about that tomorrow. We use this to, again, study Clathrometered endocytosis. And there we were interested in how molecules that are called SNX9 molecules, these little green guys there in the middle, how they are preferentially recruited towards the neck of these Clathrometered endocytosis vesicles. And so we studied that using READY in exactly the spatial temporal coordinates of these Clathrometered endocytosis vesicles and using first principle parameters. And we arrived then, after convoluting it with a point spread function from the microscope, with a synthetic simulated microscope image where we predicted how that would essentially look like, how our SNX9 would look like if we would image that using microscopes. And we essentially, our prediction panned out, and it worked. Our experimental microscopic images, they look almost exactly like what you would expect from the simulation. So that is essentially how we are integrating, like particle-based simulations with microscopy simulations. So then let me move to the other side, to the top-down, to the imaging part. So here we are using adaptive optics lattice light sheet microscopy, and I'm extremely excited about this because in my view, it is going to revolutionize the way how we do cell biology. So we used to do cell biology using two-dimensional cell cultures, then do two-dimensional imaging more or less, and then do more or less manual data analysis. And we are rapidly advancing into a world where we are creating from stem cells three-dimensional tissues. So in my hands, we essentially grew three-dimensional brain organoids. We also grew three-dimensional intestinal organoids from stem cells. And then we are using this adaptive optics lattice light sheet microscopy to image those tissues in 3 dimensions. And then when you image tissues in 3 dimensions at like 1 frame, 1 second per volumetric frame, you arrive at like data sets that are massive. It's no longer hundreds of megabytes of data, but it's hundreds of terabytes of data. And now you can no longer use, your hand and like click on and create ROIs in Fiji, but you actually have to use automated methods, you have to use big data methods, machine learning, and to actually arrive at a quantitative analysis. And so we are developing this future of cell biology right now. And I'm going to show you a little bit how we are doing that. So the microscope I'm talking about is arguably the most advanced fluorescence microscopy technology that exists. It's 100 times faster than a confocal, 100 times less photobleaching than a confocal, and it has adaptive optics. And adaptive optics really unlocks the deep tissue subcellular imaging capability. What you see here on the left-hand side, that is what a normal microscope would see that does not have adaptive optics. It's essentially a big blur where you would essentially see these individual Clathrometered endocytosis events that are 200 nanometers in size and that would essentially look like little spots. And I don't want to go into the details. This is not a microscopy talk, but the way you can think about this is, when you're in your airplane and you have these noise canceling headphones, they essentially measure the ambient noise and then put anti-noise so that you can listen to your music and don't have to listen to the airplane noise. We are using roughly a similar idea to do this in optics, where we measure the optical aberrations that the tissue induces, and we can filter the optical aberrations out by putting that on a deformable mirror to get perfect imaging results. We can talk about that in detail later, but what you can do with this now, you can actually image a three-dimensional organoid, a live organoid. So this is a three-dimensional intestinal organoid where we imaged the top part, and we labeled Clathrin endogenously with RFP and Dynamin endogenously with GFP. And then you get these beautiful data sets. So this is, the end game data set for Clathrometered endocytosis. So Clathrometered endocytosis, you could only study that with turf microscopy, essentially, where you have like one plane, high frame rates over time. Now having access to three-dimensional live microscopy and live data of the same process is stunning. So, we have access to, so this is a 50 by 100 by 100 volumetric data set where we're looking at about 20 cells at a time, where you see like all of these individual events. Those are Clathrometered endocytosis events. You see how they form on the outside of the plasma membrane, you see how they mature, you see how they get like endocytosed and how they get decoded. So everything about Clathrometered endocytosis is in this data set. The problem is it's a massive data set, okay? And we didn't really have the tools to deal with this new microscopy data because it's so rich. And so that's what we set out to develop in the lab. So when you look at two-dimensional microscopy over time, you get like maybe a couple of gigabytes of data. There's plenty of visualization tools and plenty of quantitation tools. But for our adaptive optics lattice light sheet microscopy, there were very, very few tools to visualize the data and even less to actually quantitate the data. So we developed that. And what we came up with is essentially called PyLattice. We named it for Python-based lattice light sheet image analysis. And it takes lattice light sheet data, segments individual points, tracks them over time, and then does the downstream analysis. This is also open source, GitHub/PyLattice/PyLattice. That's where that software is. And it's written in a way that you can access our library from Jupyter Notebooks, process your lattice light sheet data there. And we're going to talk about the machine learning data. But what the software does is essentially it detects all these individual spots, and then it tracks these individual spots over time. And when you then do this for the Clathrometered endocytosis data set, this is the top view of our data set, where we have color-coded these tracks for track length. So some take a little bit longer to mature, some take a little bit shorter. This is the top view, this is the side view. This is one single event. This is the quantitation of that one single event. So you have the maturation of the Clathrin code. You have then the Dynamin assembling on the neck, cutting it off, and then it gets endocytosed and then further decoded. This is the statistics about this. And then you can do this on the entire data set for the entire organoid, or the organoid section that we imaged to arrive at those parameters. So that's essentially how we are doing our top-down approach, develop these like segmentation tools, tracking tools, particle coordinates to then arrive at what's actually going on and the quantitative results here. So then the last part is the machine learning. We are using our particle-based simulation tools and convolve them with our microscope point spread function to get synthetic training data for our machine learning segmentation tools. That's essentially like one way we are using machine learning here. And so, what we're using is convolutional, like 3-dimensional convolutional neural networks. And what we found is with those convolutional neural networks, we are much better at retrieval of signal in our data sets. So with these classic segmentation tools, you really have to be high above the noise floor to actually recover your biological signal. With the deep learning tools, when you train them well, you essentially you can essentially retrieve 5 times better signal. You can like the bottom line is you can image much longer with much less laser power, are much more benign to your cells, and your cells are much happier and your data is better. So that's fantastic. It looks better. It's also faster than our classical tools because pulling inferences from a fully trained neural networks is much faster. So it works really well. So this is the raw data. This is the classical, and this is our deep learning where we can even get a little bit more out of our data sets. So that's also on GitHub. Pilot is deep learning if you're interested in that. And so this is essentially how we in our lab integrate data. So we have our bottom-up from simulation created data sets, and we have top-down with adaptive optics status light sheet microscopy, where we go top-down to essentially arrive at these system parameters. And so now I want to talk a little bit about the future and where we are going with this. So these adaptive optics lattice light sheet microscopes, they are like a new type of microscopy essentially. And so we have ways to deal with the pre-processing of the data. And I've just shown you how we have ways to deal with particle tracking problems. That's what we just developed for these Clathrometered Endocytosis studies. But for organelle tracking, membrane tracking, nuclear tracking, and all the novel biology that we're going to discover with these microscopes, this is totally uncharted territory. And so what we are now focusing on in the lab is organelle tracking. And I tell you why that is so important. And it's fantastic that we had Chantel's talk this morning. She talked about mitochondria and why they are so incredibly important in neurological and neurodegenerative diseases. And what is also incredibly interesting about mitochondria, they don't look like what you expect they look like. Okay, when you just type in Google images mitochondria, you're gonna find tons of images that look like these little kidney beans. That's not how mitochondria look like. Mitochondria are these 3-dimensional networks, and these 3-dimensional networks that rapidly change over time. Okay, and so in these changes, that's essentially fission and fusion events where the mitochondria are reshuffling their molecules to essentially stay alive and be productive in energy. If anything goes wrong with these processes, the most energy hungry tissues are going to feel that first, and that's essentially your brain. So if something goes wrong with mitochondria, you're going to see that in neurological diseases and neurodegenerative diseases. So it's incredibly important to actually really get a hold on mitochondria in 4 dimensions now that we have access to collect those data sets. And the data sets that we can collect look like this. So this has been data that has been collected on the Janelia adaptive optics lattice light sheet microscope. There is essentially a label on the cell membrane. There is a label on mitochondria. And you essentially collect the data of this 3D tissue volume, and you collect a movie of that. So then you can, after the fact, look at these individual cells from which you have essentially taken a movie from all of them. You can then zoom into these individual cells. And, let's say this cell that comes into focus here. And you can then look at the individual mitochondrial dynamics for these individual cells. And then that essentially looks like this. And this really makes you appreciate how incredibly dynamic the mitochondrial network is. And when you imagine growing brain organoids with neurological disease mutations, and you then are able to observe mitochondrial dynamics and how these disease mutations actually play out in the native tissue context, now that we collect this data, that's essentially what we are focusing on. it's a little bit too preliminary to talk about our quantitative analysis pipeline there, but it's actually ready. So if you're curious, drop me an email. Let me know. Let's have like a one-on-one conversation, or just come down to the lab, and I can show you on the computer. If you have mitochondria data like Chantel, let's talk. You know, we have the quantitative analysis tools ready essentially. So stay tuned. We're gonna publish them soon. It's exciting. And so what I have now is just, you know, like the little time lapse of us building our adaptive optics at a light sheet microscope, like just like 2 rooms down from where I am. It's operational now. So we are collecting data. It's extremely exciting times. And what I want to end with is that we have essentially an opening in 4-dimensional cell biology on the mitochondrial team. You know, it's incredibly exciting. I would like to thank my lab, thank the Hardware Foundation for funding, and I would like to thank the Allen Institute for giving me this opportunity to talk. And I would also like to thank the Allen Institute for existing, and because I am just a huge fan. You know, you're fantastic. So thank you so much, and I'm happy to answer any questions.
Thank you, Joe, for a fantastic talk. So we have time for a few questions. How do you deal with imaging thick organized like brain organized, and how do you normalize intra-organized variabilities?
Yes. Oh, that's a fantastic question. Thank you. So thick brain organoids, when you think about it, like, you know, thick is relative. Like if you're interested at subcellular resolution, like we have like a 200 nanometer resolution there, or like 100 nanometer resolution when we are using SIM, then, you know, plus adaptive optics, we can essentially get 100 microns deep into our brain organoid, and that gives you on the order of like 50 cell layers. Okay, so we can get quite deep into these organoids and still get meaningful data. It's not, you know, it's not realistic to do like adaptive optics at a light sheet microscopy and go like all the way through like a 2 millimeter organoid. No, but for the resolution and what we are interested in, it's plenty. It's plenty. And then the intra-organoid variability, yeah, that's essentially something that comes with the brain organoid system. What we are doing is essentially to limit that is to use the best possible protocols that are like, you know, turning out like very reproducible organoids. And the other way we deal with this is essentially very carefully, like when we image, like really image a region, you know, that we from immunolabeling and other things, we are confident that this is essentially like a very similar region with like the same composition, like neurons and cells in the same developmental stage. So that's how we deal with this. Yeah.
Maybe one last question from myself here. What are the typical compute resources that you need for 30 terabytes?
Yes, that's a fantastic question. Yes. Well, we have essentially wired our entire lab with fiber. There's essentially like a direct fiber link from the microscope to the fiber backbone that we have here at San Diego because of the San Diego Supercomputing Center, and you know that goes directly to the, yeah, like we have from the microscope a fiber link to the San Diego Supercomputing Center where we have massive amounts of storage, massive amounts of compute resources to help with this, and it's a problem. We can talk about this later, but you know, like since the data is so new, like all these like compression techniques or like data reduction techniques, they are all in their infancy. So we are actually working with the Supercomputing Center and many people in computer engineering to solve those issues as a team. And you know, I know that the Allen Institute is also working in that direction, and so I think it's just a team effort that is required, you know, as people in quantitative cell biology to do this, you know, because, you know, we have this amazing opportunity to collect quantitative data in 3D live. So let's get together to to quantitate those data sets. It's awesome.
All right. Thank you again, Joe. So I think, yeah, we'll move on to the next talk now. Our final presenter for today is Dr. Devin Schweppe. Dr. Schweppe is an assistant professor of genome sciences at the University of Washington. His research focuses on improving the efficiency of tools for high throughput proteomics and applying these tools to understand complex biological systems. His research interests focus on the implementation of millisecond informatics to enable intelligent data acquisition strategies. His research group applies these technologies to quantify proteins as a readout for diverse cell states with broad interest spanning microbial protein interactions, small molecule binding events, preclinical proteomics, and profiling primary tissue samples. His talk is titled Multiplexed Mapping of Complex Proteomes. Please welcome Devin.
Unmute there. Hi, thank you for that introduction. That was great. It was perfect for setting the stage for 2 out of the 3 projects I'm going to talk about. So I'm excited to be here. Really appreciate the invitation to present too. So as opposed to a number of talks today focusing predominantly on imaging, our lenses are a bit different and focus mostly on ion optics, and they're geared towards trying to understand biology through the scope of kind of proteomics, right? And so proteomics to us means a number of things. Classically, it was focused on identifications, then gradually over the last decade or 2 has moved into tracking abundance and degradation and profiles we would associate with expression, and moved into modification statuses of cells, so phosphorylation, ubiquitination, et cetera. And then over the last 5 or 10 years or so, a much larger focus has started to move into, can we understand more about kind of the proteome? So all these proteins in a given cellular system. So can we look at activity of a given set of proteins or their subcellular localization using multiplex analyses or binding interfaces and protein interactions with other proteins or ligands or small molecule drugs. And so we as proteomicists are very interested in essentially how we can improve proteomics for all of these factors. And so if we look at kind of a general pipeline that is tried and true, where we take a biological sample, isolate the proteins, digest, and then do bottom-up proteomics. One of the major caveats to this is that the actual analytical pipeline that we use is pretty much agnostic to the input samples that we have. So the mass spectrometer has spectral data that has no understanding of what the inherent biological information, what your samples are, or what tissues you're using, or what species these samples came from. And so we can begin to use quantitation to get around some of this, right? But inherently that's still spectral information. We in our lab use multiplex quantitation, which uses a chemical barcode termed TMT Pro, whereby we can take 16 and now just this year up to 18 different samples. We can label them with an individual chemical barcode, and then we can actually essentially read out the quantity of those proteins and the relative abundance across up to these 18 different samples simultaneously in the instrument, right? And so that's exciting to us because it offers a number of advantages. 3 of those just pointed out here, right? So throughput. So no longer do we have to shoot 1 sample to get that information out. We can shoot 18 different samples simultaneously, so we're at 18x throughput. We have fewer missing values, right? So every peptide or protein identification we get has quantitative values for all of the samples we input. So if we have multiple drugs or multiple time points, we can see information for all of those without losing that. And that also allows us to employ complex experimental designs, right? So we can do these dose dependencies and dose curves and look at multiple mutants simultaneously or time points. So quantitation is a huge boon for us in how we interpret the data, but still inherently the instrument is ignoring all of this interesting biological information, right? And so one of the big questions we began to ask a few years back was whether or not we could use biological information in real time with instrument acquisition. So on the order of 1 to 2 milliseconds, can we make a decision based on biological inputs? So protein sequence, peptide sequence, modification status, et cetera, to inform the instrument to better collect data. And so we began to do this, and through a number of papers and a lot of work from folks in my lab and also the GE lab back at Harvard, we were able to do this. So Using the latest round of instrumentation, we're focused mostly on thermo instruments, but this is also coming to broader spectrum instruments like the timsTOF. We're able to input real-time information on what is actually happening, right, with these analytes. So real-time search was the first major modality of this, where we can take spectral data and actually interpret what the spectra is, determine what the peptide sequence that spectra came from, and then decide in those few milliseconds whether or not that's an interesting peptide or protein for us, and should we follow up, right? And so that's allows us to do some active and exciting things, so we can spend more time on really interesting proteins, those that we know, say, are involved in a biological process or specific to our treatment set. We can also target specific proteins that we just know we want to track across data sets. We can also do things like track quantitative values across these samples simultaneously. So we can say and look for specific quantitative changes in real time and say, if we see a large change, let's spend more time and understand more about that specific protein or peptide. And so altogether, the benefits of these systems is that it allows for faster methods. It allows us to identify more proteins in less time, improves our quantitative accuracy, and also allows deeper coverage. And so all of these things kind of are under the umbrella of intelligent data acquisition, whereby we're actively using biological information, quantitative values, peptide identifications to inform how the instrument acquires new information. And so one of the big questions then is it useful, right? So you can build a nice thing, but if it's not useful, no one cares. So how and where can we apply these IDA strategies? And so I'm going to talk to you about a couple of vignettes for how we're beginning to think about this. The first is an active collaboration with the Jackson Labs, looking at aging proteomes, specifically murine aging proteomes. And then the next set is actually looking at protein-drug interactomes. So can we decipher essentially specific interactions between small molecules and therapeutics in particular with proteins at a proteome-wide scale? And so the first question relates to determination of age as a biological variable, right? So we just wanted to know how does age affect proteins and protein complexes? So it's much easier to do this on the murine scale of weeks and months rather than the human scale. And so we decided to tackle this in collaboration with Gary Churchill's group. And we collected 200 anatomical samples. So this is 20 mice, both young and old. So young being 34 weeks and old being 79 weeks old. And they're both male and female. And so in that way, we can also look at sex-based effects, though I'm not going to talk too much about that today. We collected these samples and then analyzed them using real-time search. So we're going to use that active method whereby we can match peptides and go after specific subsets of peptides or make sure that we're going after the right peptides in our instrument. Combine that with our sample multiplexing approach, and we were able to quantify over 10,000 proteins across this data set, right? So this is 200 unique proteomes. We captured this in 2.5 weeks. And if we had done this with canonical methods, it would have taken about 2 times longer. And so that, again, is the advantage of these IDA strategies. But moreover, what we're excited about is that we can actually see age-specific proteomic changes, right? So if we just look at the relative effect size across our different tissues, so you can see tissues here are rows in this heat map, and then every column is a protein. We can see specific clades within this, highlighted by those black boxes, that point to age-specific changes, right? So we can look in this adipose tissue box here, and we can see that within the fat tissue, there is a relative reduction in the presence of proteins associated with OXPHOS, or oxidative phosphorylation, and general mitochondrial energy production. And so I'm not going to go through every single one of these, but suffice to say that there are noticeable and consistent age-specific effects across this. And we can look at that in a slightly different way, on a more protein-centric way, where we can see in this volcano plot, right? So the age effect is on the x-axis and the significance on the y, and we can see essentially that some of these proteins are relatively inconsistent in their tissue age effects, but many of them, or a good cohort of them, are actually consistently changing with age across these tissues. So you can see that in some of these immune proteins, so IgKC. We can also see that across ages, some proteins have inverted responses to age. So BCAT1, for example, here in the kidney has a relative reduction in abundance and an increase in adipose tissue. Now we can take a deeper dive into BCATs. It's actually a branched chain amino acid, essentially metabolic process, or involved in a branched chain amino acid metabolic process whereby it converts branched chain amino acids to branched chain keto acids, and eventually that results in acyl-CoAs. And we know from previous work in worms and mice and other organisms that BCAT has an effect. If you knock it down in worms, it actually results in longer lifespans. And so that led us to be very curious. When we looked at kidney, we see actually a reduction in BCAT abundance. So how that correlates then to those cells and how those are essentially their lifespan and the longevity of the kidney versus fat, which we see the exact opposite trend, where we see more BCAT1 and potentially more energy production there. And so we're beginning to tease these apart, but for the sake of time, I also just wanted to point out that not only are we able to look at protein-specific effects within this background, we can look at complex-specific effects. So what is aging doing to the formation and stoichiometry of complexes? And so we can see here the CCT complex, right, which is involved in essentially protein homeostasis. We can see, I'm just showing correlation plots for young and old mice here. And what you can hopefully glean is that there's a slight difference in terms of the correlation as mice age. I think that is better pointed out in these heat maps, where we can see that the CCT complex seems to have increased cohesion and thereby increased correlation with age, suggesting that there's not only potentially more protein, but more of this complex being formed as these mice age, likely to deal with maintaining protein homeostasis with age. And so that's just one way we can kind of apply the proteomics work that we're doing. But another angle too is we can look at essentially protein drug interactomes, right? So interactomes are a major interest in our lab, whether protein-protein or protein ligands. And it takes advantage of a different quantitative strategy whereby we're more focused on just looking at the things that change in a system. And why we're interested in that is because that seems to have frozen. There we go. Right around the time we were kind of finishing up that mouse study, we became very interested in large-scale chemical library screening. And particularly what we were interested in is how small molecules bind diversely to a number of different proteins and with a number of different effects, inhibition, activation, changes in binding between multiple proteins or changes in conformation, and whether or not we could actually detect these small molecule protein interactions. And so right around the same time that we were finishing up kind of the original IDA strategy, work from Cole Trapnell was coming out in Science, focusing on this idea of doing large-scale single-cell RNA sequencing on drug treatments and titrations of various different drugs with a large cohort of them being epigenetic regulators, but also focusing on nice signaling modifiers, et cetera. And so we were interested in taking a small cohort of these that we could potentially compare in the long term and look to see if we could basically start to explain some of the effects that they saw. So one thing that they noticed is that when they treated with these drugs, they saw relatively heterogeneous responses. So not all cells responded to these drugs, one. And so, and not only that, not all that responded seemed to respond in the same way, right? So in this projection, you can see essentially that there's a very heterogeneous response within drugs treated with the same cell. And getting back to something that kind of Lucas was mentioning at the head of this session is this idea of context. So we're curious if we can add another dimension on the context of what's going on in these cells to help assess and determine why there's such heterogeneous response to these small molecules. And so we turn to a technique that was popularized by Michel Savitsky and Marcus Benchef, and it uses thermal proteome profiling to determine small molecule protein interactions. And how it does this is essentially, if you treat with a thermal gradient, we can see that proteins will aggregate and fall out of solution. And if we take the soluble fraction of this, we can actually measure how stable a given protein is. And this stability is affected and can be changed by adding in small molecules or proteins or other protein interactions that either stabilize that protein in the conformation or destabilize it. And so we can apply this essentially to ask two questions. So can we target just those things that are changing with our mass spectrometer? And can we scale this up? So a lot of these early experiments were focused on relatively small comparisons or sets of samples. And so we really wanted to know, if we're going to try and one day screen full chemical libraries, can we scale this up to a 96-well plate-based process? And so we adapted a workflow that looks something like this, where we take cells, we treat them with a small molecule, either the cells or the lysates, and then we look for thermal proteome shifts. We label those cells in either quantitative triplicate or quadruplicate, and quantify the stabilization or destabilization. And then we use our mass spectrometry-based intelligent data acquisition to specifically go after, or try to go after, just those proteins that are changing. And so we can see that this is a relatively accurate system. So we have some well-known kind of binders and inhibitors in here. So CPI-203 is a BRD4 inhibitor. And we can see nicely that when we add CPI-203 to these cells, we see a incredible stabilization of BRD4 in this system. But what you can also notice is there are other proteins that seem to be essentially bound and stabilized or destabilized. And so one of the active questions we began to ask as we assembled more and more of this data, right? So just showing 6 of these compounds simultaneously, is that what else are these molecules engaging? So what other proteins could be resulting in kind of heterogeneous responses to these drugs? And so we can nicely see for the CPI-203 that BRD4 is engaged, but also BRD2, even though this compound is thought to be a selective inhibitor of just BRD4. We can also see that if we look across this, there's a number of proteins that consistently are off-target effects. So ATP6 seems to bind just about every drug in this particular set of samples. We also see some unknown targets in here that we can follow up with. But the engagement, right, of the off-target and also potentially on-target effects could help us likely understand these heterogeneous responses. And so we'll be pushing that more and more over the next few months and years, with the ultimate goal too of combining that back with these single cell RNA sequencing data to really interpret how these effects are shaping the cells. So basically at the end of this, I'll just summarize by saying we're able to do 96-well plate-based assays, but this is still not really at full speed. So we can do 96 compounds. We can do this faster than folks have ever been able to do this, but it still takes us 12 days to go through 96 compounds. So we could only get to about 3,000 compounds a year. So we can and need to go faster. That's both a combination of instrumentation and sample preparation. But we have an enthusiastic yes for both of these. I know both postdocs working on this, we're very excited about that, but we can do better. So with the last little bit, I just want to thank the folks that did the work, particularly Megan and Jonathan for the work on small molecule work, studies, and then also Greg, who has been leading the bioinformatic charge on the mouse work. And I'm happy to take questions.
All right, thank you, Devin. So we have 2 questions in the chat. First, can we use this directly in C. elegans? Maybe it will be easy because of its transparency.
So so we can use it in any biological system. So you can target any organism, anything you want to, for the proteomics work.
Okay. And one more question from Gokhan. Age-specific proteome is very interesting. Can you stop or reverse aging proteome either by drug or diet interventions?
We would love to know that. Yeah, that's exactly where we're trying to understand. So the big kind of start is just figuring out the major pathways kind of that are affected. And those 2 seem to be metabolism, or I guess 3. The main pillars are metabolism, proteostasis, and the immune system across all of these tissues. Those are the consistent responses, and figuring out how any one of those tissues responds to age, and then potentially finding a way to ameliorate those effects. Yeah, it's a great question.
And one last question, can combination of drugs, and this is by Brock, can combination of drugs by matrix somehow solve your coverage problem, or does the data become uninterpretable?
Yeah, no, that's a great question. I think the data would be hard to interpret, but not necessarily impossible. You can imagine if we can get an idea at least. So there's a large-scale MOA study of proteomes that's coming out, I think later this year that we worked on. There's also large-scale cysteine profiling. And so we can use those other large-scale proteomics data sets to get an idea of drugs that are likely to give similar profiles, and then likely backtrack that to make libraries where we could do kind of these kind of matrixed setups and hopefully combine 2, 3, 10 drugs per well. So it's a great question and a great thought. And we're kind of working to see how best to do that.
Okay, thank you again, Devin. So now to conclude today's session, we're going to have a panel discussion with the speakers from the day. Members of the symposium committee crafted these questions to discuss the speakers' experiences and challenges with data sharing and integration and building successful lab environments. We're going to ask all the speakers to join us on camera now, and I'll ask that Gokhan returns to moderate the panel. Thank you.
Okay, thank you, Ritvik. Now that I see all our speakers here today, I would like to thank you all of you again for these great presentations. We will start by asking first question to all panelists. The first question is, what do you see as the biggest challenges in the field of data integration? How can the field come together to address them? Can we start with Joe?
Yes, of course. What do I see the biggest challenges in data integration? You know, from my perspective, and I highlighted that a little bit, we are really in the age of where new instruments churn out incredible amounts of data, and we don't really have the analysis tools yet to really like deal with that. And so I think it has to be a massive team effort of you know, where we as a cell biology community and as a computer science community like reach out to also like across departments to essentially talk to computer scientists and say, hey, we really need dedicated support here, and also reach out to funding agencies and say, look, like I have the ability to create incredible data sets, but you know, if you want to hire a computer scientist, they have the ability to go to Apple or Google. So you have to essentially pay them the salaries that they will get in industry to make their time worthwhile. You know, so I think it's just incredibly exciting. You know, like it's the time where we really need to talk about data integration and do it right, and it has to be a team effort from my perspective. So.
Great. Thank you. I'm going to go left to right on my screen. Next is Devin. Devin, would you like to give your opinion?
Really, I would totally agree with Joe that getting phenomenal buy-in from titians is very hard these days to do that work. I also think kind of from a topic or field specific, one of the big things for proteomics is that a lot of the data is bespoke for a given experiment, and getting that into kind of standards, not of raw data release, but of quantified data analysis, is still something the field in general is working towards. And until it really like accomplishes that efficiently and well for just about every lab, it's going to be really hard to kind of integrate that with completely wholly different omics sets. So, yeah.
Yeah, I agree. I think the scale of data coming in is a big challenge to think about, just how to handle the infrastructure of it, and maybe even a bigger challenge to know what to look for and how to really take advantage of what's coming in. And I think that's going to be tricky going forward, right? Because you can imagine you're getting terabytes or more of data from some assay. Like we also do Lychee, ton of data coming in. Probably the most straightforward way to think about it would be we come up with an analysis pipeline that makes sense to answer some question, try to extract the information out of that, and package it in a way for a publication or for a story, and then archive, and so that's a reduced footprint format for the analysis. Then we archive the raw data somewhere, hopefully after that. But then once you've done that, you've kind of, unless you think to go back to it or know how to go back to it, you've then lost the opportunity to explore the feature richness of it, right? So that knowing how and when to interact with it and deal with the storage and migration all at once, I think is, especially as you start to go across multimodal information, not just imaging data, but then integrating that with sequencing and everything else, mass spec, it's just going to be a whole new world in that sense.
Thank you. And next, Chantel.
Yeah, I guess I would just echo what a lot of people have said. I think right now we're at a time of, with microscopy advancing at the rate it is, we can see things that we've really never been able to really see or quantify before. And wow, that's very exciting. It's like once you image that and you have all this data, it kind of is a daunting task of where do you begin and how do you set up that analysis workflow? And also how do you integrate, a lot of the data scientists to, for a long time, I feel like biologists have been so siloed and, we've been able to quantify what we need, but now we really are relying on the data scientists. And so it's coming to a point where we've used so many, I guess, like kits or simple ways of you click this button and then it does this for you. And that has worked, but now we're moving to a point where you cannot do that anymore. So it's also, we have to think about training the next generation to have all of these skill sets moving forward. You can't just be a biologist anymore. You've got to be multidisciplined in order to answer a lot of the questions that we're being able to ask now.
Thank you. Last but not least, Lucas.
Yeah, I of course agree with what was said, but I think if you look at, let's say, physics, right? I mean, physics is able to integrate everything by trying to go down to standard physical units, right? With that, you have standard units with which you can describe everything. And we are still very far away from that in biology. And that's going to be the biggest challenge, right? But if we find common units that can be used in all types of measurements we collect or derive standard units out of them, then of course we can do a lot of integration. But I think it's a huge challenge, of course, right? Yeah.
Go ahead, Joe.
I wanna say something to what Lucas just said. I think Lucas, you're totally right. Also in the aspect that we are not, like, this is not the first time that the problem like this has happened. Like the physicists have solved this problem. In biology, this has happened multiple times. Like the whole field of bioinformatics is essentially born out of the automated sequences haven't been invented, and now people no longer do manual Sanger sequencing, but you essentially get massive amounts of genome information from these automated sequences, and that's the time when bioinformatics has been invented. So and so we can learn from those models, and I think it's essentially very rewarding to go back in history and say, okay, what can we, like, what's different now, what can we learn from other models, and then get together as a team to address those things.
Right. Yeah.
Any other thoughts on this before we go to the next question?
I mean, of course, essentially in seq data, it's four letters, right? Literally, I mean, right? Can be a bit more, and then you can have modifications and all that, but it's still, in the end, the unit of measurement or of type of information in those data is much smaller. The diversity of that, then, think about all the things that you guys have presented, right? That you can measure now with live imaging, multiplexed imaging, looking at all the different dynamics, properties, numbers of organelles, shapes, all these properties, right? I think it's still a huge challenge there, that where bioinformatics, let's say, of this type is I'm not sure it's going to help us a lot there, to be honest, but maybe it does. Yeah.
Maybe the closest related example is not really bioinformatics in the sense of sequencing bioinformatics, but it's more like bioinformatics also in the sense of like what happened with X-ray crystallography. Same thing. Like at some point you had the ability to create X-ray crystallography structures from to get X-ray images, right? And then you essentially had to develop the computational infrastructure to go from the diffraction pattern to a structure.
Definitely.
That was essentially like now you're no longer talking about like 4 letters, but now you're talking about, again, the physics of like a diffraction pattern and how you can get that into a molecular structure. And it's a similar thing happened in cryo-EM, like where you essentially go from like individual and essentially like building these tools like Relion and other tools to essentially make the current single particle and cryo-EM revolution possible. So I think what I'm saying is maybe the 4-letter data revolution is maybe a little bit off, because we have more complex data now, but it's there's more to learn from, I think. That's what I'm gonna say. Yeah.
Hey, if there are no more thoughts on this, I'm going to start asking questions to the speakers, individual questions. And I'm going to go again on my screen from left to right. Joe, first question is for you. How do you advise the people in your labs to deal with the stress of the publish or perish academic life?
What I do is I'm essentially I try to do 2 things. I try to be like extremely upfront about that, and that's essentially just a reality. At the same, and then I think that like a holistic approach to like PhD advising is really important, which should also include, like mental health and like that one takes care of oneself in a holistic way. And so we are talking about it all the time. So it's because it is a very stressful environment. And so I think the good thing is we are starting to see a process in academia where academia actually starts to open up to these processes that is actually a very stressful environment, and where conversations about, let's say, mental health is actually not like a taboo topic anymore, but you can actually even say that word, like say in a panel like this, and so I think that's actually very important. And so yeah, like I think what I want to say is like like it has to be a holistic approach to training the next generation of scientists. Just as Chantel said, we have like it's incredibly important that we train the next generation of scientists well, in all of these new tools, and that those people are the future, and we have to give them all the tools and everything that they need to be successful.
Thank you.
I like that. Can I just add to? I'm curious because I think one thing that's helped me, and one thing I think like Ben Garcia in proteomics is doing recently, is like showing that success is not a monolith, right? There's a lot of ways to be successful as a PI. You can leave academia and get out of publish or perish, right? And I think there's more and more examples of this that are more tangible for folks on either Twitter or other social media outlets. And I think that's a powerful means to feel connected and not isolated in such an environment.
I'll also add that I feel like more and more people are showing their failures as well as their success. I think for a lot of students, they feel that these top scientists are just, they get grant after grant, all papers get accepted, and that's just not the case. And so the more that, people who are leaders within the field are honest about their successes and failures, I think that will show students that it's okay that I didn't get this, I'll get the next one.
I love that. So that's like Ben Garcia just said he got rejected from I think all 14 grad schools he applied to, and now he's like the head of a department in St. Louis, and you're just, yeah, that's awesome. Totally agree.
Anybody likes to chime in? I think our listeners can benefit from your experiences and your thoughts on this topic, especially.
I agree with everything that's said so far. I think it'd probably be a little bit naive to think the goalposts are really moving, but it is interesting to see now that preprinting is becoming more accepted that, maybe we're at least thinking a little bit more about what these goalposts mean. The way we try to think about it in terms of generating a study in a lab like ours is do good work, write a good preprint, put it out, and we'll play the game with the journals we have to play, but we have some agency to kind of control at least the scientific destiny of it. And the fact that so many groups are willing to share at that stage, as opposed to pushing it back to who's getting the publication slots in these specific journals, which obviously that game is still being played, but I think it makes the process at least a little bit more visible than maybe it was before, or at least it did for me, kind of as an earlier career scientist.
Yeah, definitely. I agree with that fully. I'm sitting on quite some of these panels that recruit new faculty, and at least on the ones I've been sitting or am sitting, I see quite a big change in how we are looking at candidates. I definitely see here in Europe for sure that we are much less looking at journals. Like which journal did the paper appear is less of an issue and becomes even less an issue. I think it's slowly fading away, but people really take time to look at your work carefully, and I think that's also, for the young people who are looking for faculty position, I would always advise, of course positions are limited, but you want to also be somewhere where people take your work really seriously based on what you actually did in terms of the science, independent of in which journal it appeared, right? And I think people are appreciating that more, and I find this a very good trend. I'm very happy about that. Yeah.
Thank you.
There might be one additional thing, maybe. What I find is also changing is, I think it used to be that if you're in academia, then that going to industry or to other jobs is like a taboo. And I've seen I was like in industrial outreach programs in Berkeley, and then people would come to me and tell me, hey, don't let my PI know that I'm actually like in the, like that I've been here, that you've seen me here. And it's like, and I think that is rapidly changing. Like I think we used to have that in the past, but now it's a perfect, like to me, it's a perfectly viable approach to, like, be a grad student, then go to industry, and you can do fantastic work in industry, or come back. There's no, I think you have to do what is right for you and your career and what you want to achieve in life, and so that is also like one of those like old taboos that I think is rapidly changing to the better.
Right.
Thank you all. Next question is for Devon. What are the current challenges from your perspective on sharing data with the community?
That's a great question. I think making it part and parcel with the culture of the field. I think, again, field specific for us, proteomics is stinted in terms of their response to making their data available. If you're forced to do it from a journal, people do it. If you're not, people don't. Making that an inherent part of the culture to share, that you're excited to share so that people can use that data later on. I think it also comes back to some of the points brought up in the integration portion of things. It's if there's a viable means to use that data in the future, people will submit it so that their data gets used and referenced and people can build off of that. But until there's kind of a culture for data sharing, that's a real challenge. I think there's also, until it can be challenging too, that, and this came up in another task force that we were working on, which is this idea of how do you accrue metadata, right? So how do we make some proteomics sample viable for, say, the other panelists' labs, right, to build off of? How do we make it so it's useful that you can come and see 10,000 proteins and make sense of that and use it for yours? And having an appropriate stack of metadata to make that viable is really challenging, right, to come up with, but really essential for sharing that with the community.
Anybody wants to chime in also? Go ahead, Joe.
I think what Devin says is perfectly right. I want to add to. I think that there are two things required. One thing is, like the culture to change, and that is changing to the better. But the other thing is also the ability to change from a monetary perspective, because it's cloud storage is not for free. And it depends a little bit on what you want to share. But if we want to share 3 terabytes of data per hour becomes quite a meaningful amount of data when you essentially want to put that somewhere on a cloud storage server. And even the Glacier, where it's like not really accessible, is still meaningful amounts of money when you talk about like thousands of terabytes. So I think that it's, again, I see this as a kind of like a community building and community decision type of thing, where we have to, like, as a community, come together and both as the researchers, but also as the funding agencies and everybody in the community, to realize, okay, if we want sharing, then let's incentivize sharing by like everything, and that also includes the money to pay for it.
Well, in regards to that, Joe, what type of platforms do you think people can use to discuss these points? Is there an actually specific platform to discuss?
Actually, I think the Allen Institute is like a fantastic pioneer in that regard. We have just recently we had a big panel discussion, like, with funding agencies, researchers in the field, and people who are like pushing the field forward to, like, get everybody in the room and say, look, this is this current status, this is current costs, this is how this looks like, and this is the mentality that we want to address and we want to change to the better, and this is how we might want to pay for it, and if we all agree that this is exactly what we want to do as a field. And so, I'm a huge fan of the Allen Institute, and because it can be the nucleator of those conversations. Because at the end of the day, like, when you say, am I going to like be like as a just assistant professor, a small PI somewhere, am I gonna say, like, am I gonna by myself change the politics of US research funding? Probably not. But if we're gonna do that as a team, then yes.
Thank you.
I think it might be interesting to also look at it from the flip side, right? Like, how many people are using, I guess, going through the literature and using someone else's data to reinterpret or reanalyze to come up with different scientific questions. There are some data sets that I think are extremely useful, and there may be some that may be less useful. So to have the requirement that you must put it up there, are we just polluting the data by putting too much information out there when it's making it more difficult for us to interpret that? And also the data that's going out there, are you putting it out there in a way that someone else would be able to use it and really interpret it? I think are things that we need to ask ourselves and make it more user friendly, because otherwise people aren't going to use the data sets that are available.
Right. Yeah, I fully agree with that. I think it's a great point. Data, I've been in the business of putting large data sets out since a while. And, honestly, the data we were collecting, large scale, multiple terabytes of data 10 years ago, it's not useful anymore, right? I mean, the technologies are way better now. And so the speed with which you could collect those data back then and compared to now, it's a fraction of the time you need now. So, data gets old, right? And then, they're not useful anymore, right? Certainly in our field. Yeah.
Thank you all. And next question is for Brian. How do you identify new avenues for discovery?
Oh, That's a broad question. read a lot of papers. Yeah, I don't know. I mean, go to things like this. I mean, just if I want to give a broad answer to your broad question, the most engaging thing besides getting to work with my team every day and work with the students in our department and the other, my colleagues in the department at the university is just to have a chance to see what other people are excited about and kind of hope I might get lucky enough to have that moment of insight where, oh yeah, it would be really interesting to match this tool that we're working on to this unmet need that I never would have thought of, because we were able to bridge this connection or to synthesize that two data sets that seem disparate might actually be more related than we thought. I think Twitter has also been great for that, at least for me, especially during the pandemic. I mean, it's harder to get to things. There are online events, which is nice, but there's always a Zoom fatigue to kind of battle, and just having that steady stream of people kind of sharing what they're excited about, sharing their accomplishments, especially is a great way to keep that kind of going.
I think paraphrasing too something Chantel said, right, which is you have to have like a foot in multiple fields, is step outside of your fields and try and look back with some new perspective. I think that's helpful for generating ideas of what you can do within your own work, which I think relates to what Brian was saying, go to meetings and see how people are doing that, especially meetings like smaller meetings, Gordon, Keystone, et cetera, where you can see ideas in a kind of related field. That can be really helpful. I think it's also, there's whole weird projects that we've got into just from simple emails and a question that got posed that spiraled. So I think one, being open, but two, also having the courage to reach out to people who are almost always really excited to work with you and make these ideas possible.
Yeah, I think sometimes within fields, you can just start all doing the exact same thing. There's one leader and everybody's just following, doing similar research, asking similar questions. And so it is really nice to always step outside the field, talk to someone who's in a very different field about your project. They're going to ask you either really basic questions that you thought you knew the answer to that you don't, or they're going to make you think about things in a brand new way. For me, that's going to the neurobiology department. I'm always worried about what's happening within one neuron, and they're like thinking about how networks are talking to each other and all of this. So having those interactions, I'm like, whoa, okay, I have never thought about this from this way. And so I think those are what you need in order to have like a good collaborative environment where you can seek out those resources is always great to have.
Yeah, I try to tell people, go out of your comfort zone, right? Go really out of it. I think that's sort of key. I like to do it myself, and it triggers new ideas. I think it's really not good to stay in your comfort zone. Right.
I think to me that includes, like as Chantel said, like talk to the neurologists, but also talk to the data scientists and the computer scientists and the physicists on that side. But also like I talk to clinicians a lot, actually, because they might actually have a patient with a incredibly interesting mitochondrial disease that nobody has looked at so far, and it would be fantastic to use like the most cutting edge tools in basic research to figure out what's going on, and so yeah, like expansion of your horizon like that is, I think, a good idea.
You all, and next question is for Chantel. What do you feel is the biggest unsolved mystery in the field?
I feel like somehow everybody else got an easier question than me. Biggest unsolved mystery. Well, I think it depends on what field you're in. And so I guess for me, being in like the neurodegenerative field, I think there's a lot of unsolved mysteries about, how and why people develop certain neurodegenerative diseases. For me, what I always find really exciting is in the pathway that I study, 2 proteins are implicated in Parkinson's disease and 2 are implicated in ALS. It looks to be like some very linear pathway, but how is that even possible, presenting with 2 very different diseases that are affecting 2 very different neuronal populations? You know, other things within neurodegeneration, I just find fascinating and very confusing about how certain populations are affected by different things. I guess that's me coming from my very neuron-specific point of view. But, you know, like, take basic questions. Why do we age? How do we age? Something as simple as that seems like, of course, we have the answers to that. But as soon as you dig deep, you find out that that's not the case. Or, you know, how does your brain really work? We don't know that. You know, so those really more existential questions. I mean, will we ever get there? I don't know.
Any other opinions?
I like that. I like the particularly the forest for the trees kind of mentality of those big questions, and are you actually answering those? And I think similarly with kind of the aging project, we're looking at what is actually happening. Yeah. But also, I think when we think of large cohorts of proteins, one of the things is how, like, if we measure everything in its own little island, and this protein A goes up by twofold and protein B goes down by twofold, how those things relate, and really making sense of that data. And I think getting to earlier questions, assimilating that with other data types that actually can offer orthogonal means, like the means to do that. So part of the questions here are still the big questions in kind of our field. So.
I think what Chantel and Devin said is just at the core of what needs to happen. Like, and actually what the core of this panel is. It has to be data integration. You know, like bio is so complex that, you know, like you need the proteomics and you need the imaging and you need all of those information sources together to then really be able to say, well, a twofold difference here and a like threefold difference there together in that massive context of what biological complexity really is in that particular cell type, in that tissue. That then leads to neurodegeneration. And so, yeah, I think it is actually, you know, like the challenge is the complexity of biology and how to deal with it. And data integration is maybe the solution. And then I hope, you know, I'm an optimist by nature, so I think we can get there. You know, like we have, you know, the computer now that can essentially store all of these pieces of information. We just have to like engineer solutions to get there. So I think we're gonna get there, hopefully.
There is no other opinion on that. I'm going to ask question to Lucas. How can we interpret and integrate data coming from different model systems?
Right. I mean, yeah, I mean, again, if you are able to have some standardized set of units, I mean, then of course you can in my opinion, very well compare, let's say, a system you study in a mouse embryo compared to zebrafish. I mean, that's still maybe even relatively comparable systems, or even you want to go to yeast, that's maybe a bit far away, but I think you can do that. Another thing that we are currently sort of intrigued in, but obviously here I'm obviously biased by what we're doing right now in my lab, is this trying to approach it from the concept of information. So the cool thing of information is that it doesn't need to be, it doesn't have to have a unit. It basically looks at variation of measurements, and then it looks at mutual information between two types of measurements. And I think that allows you to sort of quantify without the need for any units to which extent there's mutual information. And then you can sort of start thinking that, okay, this is also how you can cross scales and so on. And I think from that, you can see patterns that, you know, you might see that in a mouse embryo and in a slime mold system, right? And you might see that from a mutual information analysis point of view, you might see very similar patterns. Maybe you see very different patterns, right? But that's perhaps comparing things without the daunting task of having to define everything in physical units, even though I eventually think we should try to go there, but that may be not realistic in the near future, right?
In that regards, can I actually ask, because you mentioned that, you know, similar patterns in different organisms, right? But can similar patterns in different organisms have a different outcome in different models?
Yeah, it's of course hard to say. I mean, what I'm fascinated by is when within the limited scope of my own research is that, you know, you start seeing concepts like multimodal perception or contextual information processing is actually something that has been much more developed in fields like ecology and neuroscience, right? And so these are totally different skills. Yet at the conceptual level, I think you can see very similar things within single cells. So even, of course, the details will be different. I mean, are by definition very different, right? You're talking about multiple neurons integrating in some neural network versus multiple signaling nodes in a single cell. It's, of course, by definition very different, but conceptually, they might work according to similar principles, right? And I think that's what eventually we want to learn, right? And of course, if you look at an organism that has a certain trait and phenotype, then of course the states in which the cells of that organism are in may have very different properties, right, than of another organism. So of course the details, is it the early endosome, or is it the lysosome, or is it the cytoskeleton? I mean, those will be different, but the principles might be, well, that's my hope, might be surprisingly similar in the end, right? Yeah.
If there are no others, anybody wants to also chime? I mean, I allow this, you know, engagement in here, but we also have, you know, time limitation. Maybe one more person. Anybody wants to give any opinion before we go to the last question for the panel?
I thought Lucas's summary was good. So.
Okay, I love it. Okay, so this one is for all panelists, and feel free to chime in right away. How do you envision different layers of information being transmitted across different scales? For example, how can we connect molecular information to cell information to tissue information?
Well, I think I think I've essentially outlined that in my talk, how we're doing that. I think the challenge is we don't have the computational power or the information to really like link all of these different scales to each other. But you can make abstractions. You can essentially take and that, you know, you can take a QMM simulation, then go next step to an MD simulation, then do a mesoscale particle dynamic simulation. And then, you know, you essentially, like with QMM, you get like, I don't know, nanoseconds, the next you get milliseconds, then you get seconds, time scales, but you essentially have to shave off like different layers of information. And so then from the mesoscale particle simulation, you can then hopefully get like some kind of like transfer function to like what we can see in the microscope, and to then get to like real biology. And luckily we have now microscopes that are able to actually acquire subcellular resolution in tissues. So the adaptive optics will help to really like see what's going on with different cell interactions in the same movie. So that's at least one way from like a small way how this could be done.
I think also in terms of kind of that scaling, I think one interesting thing is, for a long time, I think there was the focus on just more of everything, right? More genes, more proteins, more analytes, and rethinking some of those data sets to look specifically at subcellular structures, right? So big thing now is, can we build up kind of landscapes across a cell or population of cells of where proteins are at any given time. And that's naturally something, right, that bleeds into great collaborations with imaging labs, right? Because it's like, okay, we think these 5 proteins go here. Do they? But then we need imaging to show that. And then like essentially we add structure and layer every time we kind of add those experiments. So we can see globally what proteins are doing, specifically what these kind of localizations are doing in subcellular areas. And as I think the easiest way for us and for me has always been collaborate with people, and that'll add a layer of new information and structure and detail. Whether you go up in terms of more proteins, more cells, more organisms, or down in terms of one protein, one pathway, one drug. That seems to be the simplest.
Maybe I can add something very, very quickly. So what Devin said there is super important. You know, like you said, hey, we have information how these proteins could go somewhere. We have to collaborate with an imaging lab. You know, as imaging people, we have to collaborate with you know, proteomics and genetics labs, because you know, at the end of the day, we can maybe hope for 7 channels max. You know, and so you know, there's nowhere near like all of these like different multi dimensions that you can pull out of like a proteomics experiment, but they are all there. So it's super important to like not forget that and essentially like come from both sides and like connect those.
Yeah, I tell all of my students with live cell imaging, we only see what we want to see, right? And that's a wonderful thing that we can see it, but it also is a limitation of our experiments. So it is very important to collaborate with other people. And I think also we're reaching a point where, you know, everyone used to be very siloed, and having a lot of collaborations wasn't seen as like a positive thing. But I think now as we continue to move towards these larger data sets and acquiring all of this information, that we're finding out you can't handle it all by yourself. You have to collaborate with, you know, the people who are doing the proteomics and, you know, other people in order to have a more holistic understanding of the question with which you're asking. So I think that's how we start being able to build from system to system. Continued collaborations, continued talking, continued data distribution is how you get there.
Right. Yeah, I think, if you would go from the very extreme, let's say, sort of the most detailed scale of measurements in biology, that's more roughly structural biology, and then you can go to whole organ imaging, right? And then it's quite, or some people sort of think that you would need to have that level of detail of structural biology across your whole organ, right? And I think I'm not sure I would agree fully with that. I mean, we can coarse grain a lot as we cross the scales. Now we still need to learn how we can do this in the best possible way, but I think that's exactly the challenge, right? To learn coarse grain across scales. We are also not considering, the quantum behavior of electrons in atoms typically, right? We just fully ignore this, right? Because we don't need to go to that scale. And so I think there's hope. And also I think in the end, a lot of biological data is sparse in information, right? I mean, there's a lot of dimensions you can reduce, right? There's a lot of sparsity. So that also gives us hope. And I think particularly machine learning can help us a lot there. Right.
Hey, if there are no more comments on this, I would like to thank all of you, our speakers today, for your excellent presentations and further discussion in this panel. I really enjoyed it. Thank you for your inputs. And thank you everyone for joining us for day 1 of the Seattle Cell Science Symposium and for your engaging questions. This concludes the webinar for today. We look forward to seeing you tomorrow for day 2 of the symposium. We welcome you to review the agenda for tomorrow via the link in the chat. Have a wonderful rest of your day. Thank you. Goodbye.
Thank you very much. Bye-bye.
Bye-bye.
Thank you.
Bye.
Bye.